Okay, so welcome back, everyone. We'll be resuming with the last session of the conference today. We've been calling this the Celera session, although Celera is kind of mixed in throughout all of these. There is a change in the program. Some of the online people could not move the time, so we're going to be moving Gene Myers later in the program, and we'll be picking up right after what's listed as the audience Q&A session with Pavel Pevsner. So gives me great pleasure to introduce Pavel Pevsner, who probably doesn't need any introduction here. He's one of the founders of the Recom series with Soren and Mike, and really one of the seminal contributors to everything sequencing that people work on today. So let me just step aside and let Pavel tell you what he wants to talk about.
Thank you, Russell, and happy birthday, Soren. I apologize for not being able to join you and esteemed guests in person today, but if I were there, I'd definitely sit next to you, especially at the conference dinner. Let me explain why. Back in 1995, when Soren, Mike, and I started talking about a new conference, the term bioinformatics was still new, and computational biologists didn't have a conference to call their own. Soren and Mike didn't really care where this conference would happen, but I was a skiing fanatic at that time, and I insisted that Recom should take place near a ski resort. That is one of the reasons why the first Recom took place in Santa Fe. The third Recom took place in Lyon, where I hit the slopes in the Alps. But as thrilling as skiing in the Alps was, my most cherished memory of this Recom was the dinner at a renowned Michelin star restaurant. It was held in a palace with a waiter behind each guest. And this was where I discovered the true benefits of sitting next to Soren at a dinner. Shortly after the first dish was served, Soren casually asks the waiter if they have some potatoes in the You see, Soren's culinary preferences are quite basic. He enjoys simple Romanian potato dishes. I thought the waiter will consider it an insult and show us to the door, but he didn't even flinch. It's Michelin-style training. Within minutes, Soren had a plate of boiled potatoes in front of him. And that, when the true benefits of friendship and sitting next to Soren truly materialized. Thanks to his simple tastes, I might have been the first person in this restaurant to ever get a double portion of Michelin star food. Because Soren refused to eat his course and ate potato instead. Fast forward to the next year Recom in Tokyo, where I found myself once again sitting next to Soren at the traditional 10-course kaiseki dinner. It was a serious multi-course affair. Most people in Japan had never tried a traditional kaiseki dinner. Soren, always up for culinary adventure, attempted the same potato trick. Well, it did not work in Japan. The vendor just smiled politely, just another weird joke by a crazy American. I suspect Soren might have been the only person in the world who got hungry after a kaiseki dinner. Yet, sitting next to Soren, I got to enjoy a double portion, make it a once-in-a-lifetime experience. Those were the days, Soren. Inspired by that dinner, I want to conclude with a simple haiku. Kaiseki dinner de kueo tachi, ichire wa manpuki, manitori wa nanaka gasuit imasu. And the translation is, all friends at kaiseki dinner One is full, the other is hungry. Thank you, Florian, for delicious memory and for always keeping us entertained with your culinary adventure. And here's to a memorable moment in the future. And may your love for potatoes never wane. Oh, I forgot. You asked us to present an open problem, right? And here's a photograph from Tokyo. I believe right before we had this kaiseki dinner, and my talk will be on the recently emerged problem of sequence alignment. Now, Soren, you asked for new open problem, but roughly at the same time we were talking about recomp, Mike and I published a paper with 56 open problems. Most of them remain unsolved, so I could simply bring one of these unsolved problems to your conference. But of course, I know that you want a problem from one of your heroes. And I try to come up with something that is similar to the famous Solon's problem. What is the length of the longest increasing subsequence in a random permutation? And of course, this problem is sequence alignment, and it resulted in very deep, hundreds of deep mathematical results related to bioinformatics and not related to bioinformatics. Now, some of us, I myself recently considered alignment-based sequence comparison as a solved problem. But as Poincaré said, there are no solved problems. There are only problems that are more or less solved. And over 20 years ago, you participated in sequencing of the human genome. But of course, this sequence had thousands of gaps. And some of these gaps were in very complex and biomedically important regions. 20 years forward, the entire human genome was formed. Was finally sequenced. Well, but it is just the beginning. After genome is sequenced, different genomes have to be compared, and it turned out this alignment, genome scale alignment problem is a challenge. The first person who compared two human genome, actually two human genome assembly, was Soren back in 2004. So it looks like we have already a solution for these hundreds of new complete telomere to telomere genome, but it turned out it is not the case. And the reason it is not the case, and we need no new algorithm, is because these extremely complex regions defy all existing algorithms for sequence alignment when we try to compare them. One example is centromere, which is evolutionary enigma and still unexplored area for analyzing genetic diseases. Another, one of the most biomedically important regions in the human genome, immunoglobulin loci, we still do not know how to compare them. And another thing about centromere, they're fascinating. Since there is no recombination, your maternal and parental centromere on chromosome 1 may be very similar because they had a common ancestor, let's say a couple of 100 years ago. Your centromere on chromosome 2 may be extremely different because their common ancestor lived tens of thousands of years ago. And that makes their comparison very difficult. And that's why the first paper from the Human Pangenome Consortium basically stated new alignment methods may need to be developed to fully understand and characterize this component of the human genome, and existing alignment methods, as beautiful as they are, fail for any choice of parameters. So we started to address this problem with my student Andrei Bzikadze this year. And as an example, if you look at, let's say, two first complete centromere on chromosome X from two individuals, it looks like there is kind of nearly perfect alignment of them. This perfect alignment has nothing to do with reality. The alignment, in fact, reveals very complex evolutionary history, and traditional alignment and correct alignment actually have no points in common in this alignment grid. So something new needs to be done. And so why alignment methods do not work in this area? Let's say if you construct a dot plot of all 100 maps in centromeres, then it will not reveal any similarity. Why? Because most 100 maps from human centromere occur over 1,000 times. So there is no useful information in this dot plot. What we notice that instead of comparing all k-meres, we need to compare only unique k-meres. And if we compare unique k-meres, the similarity between these two centromeres suddenly revealed. What we later realized that fixing the size k would not work, and instead we should look for unique substrings that are shared by two genomes, and then we are able to construct alignments that reflects evolutionary history. So given string as a T, a substring of them is unique if it appears exactly once in each of them. And these are the very valuable elements of true similarity between strings. Some unique substrings may be short, some may be long. We need to combine them somehow. A uni-alignment is simply an alignment using unique substring as the atomic units for scoring. We don't use anything else. And immediately, two questions arise. Are uni-alignments more biologically adequate than standard alignments? First question. And second question, can we efficiently find unique substrings? Efficiently means very fast. Or the subset called unisets, so that the uni-alignment is faster and more accurate than even the fastest implementation of alignment algorithm, and even Minimap. So these two questions are not questions that I wanted to formulate for you, Soren, because they were solved this year. The answer to both of these questions is yes. You can design more adequate, more evolutionary adequate algorithm for comparing highly repetitive regions, and even whatever regions, even regions without repeat in the human genome, if they are not too diverged, that is more accurate than alignments, traditional alignment, and it is blazingly fast. Why it is blazingly fast? Because it's combined combinatorial pattern matching, two key ideas from combinatorial pattern matching, longest common prefix array and sparse dynamic programming. Since both run in nearly linear time, the result, and since the number of unisets is small, the resulting uni-alignment algorithm is blazingly fast. Now, back to Ulam's problem. Back to Ulam's problem, how the problem of uni-alignment is related to the Ulam problem. I mentioned that some centromeres of whatever highly repetitive region in human genome are quite similar. Others are extremely different. These differences are not really apparent. They only become apparent when you apply uni-alignment and look at unique substrate. So this is centromere 12. We suspect that the common ancestor of this centromeres lives tens of thousands of years ago, some of the most diverse centromeres. Basically, the alignment mainly consists of insertion and duplication rather than matches. And the question arises, when alignment is very diverse, it becomes statistically insignificant. And when uni-alignment is that diverse, it's probably statistically insignificant. How we define this gray zone where uni-alignment becomes statistically insignificant? Now we come to very deep mathematics. If we consider random dot plot and point randomly uniformly distributed at random in the unit square, and define A of n as the scorer of the optimal northeast alignment path with its maximum number of dots, then we can, if we learn the distribution of this random variable, then we can say something about the scorer of uni-alignment. If a uni-alignment scorer does not significantly exceed expectation of AN, it indicates that unisets are randomly distributed in the alignment grid, and the uni-alignment is statistically insignificant. Thus, to get a key for evaluating statistical significance of uni-alignment, we need to get a key on the distribution of AN, And this problem was brilliantly solved actually quite some time ago, because the random variable an is closely related to the solution of the Ulam problem. And Ulam problem was solved by Logan, Schaap, Vershik, Karov theorem, and who proved that the length of longest increasing subsequence grows as 2 square root of n. Now, with this at hand, we actually designed a rough empirical estimate of the statistical significance of a uni-alignment. It's in our recent paper published this year. But this is a poor man's solution to a complicated probabilistic problem. So the Ulam-like problem for uni-alignment, what is the score of the uni-alignment between two random sequences? What is the expectation of the score? And this problem, of course, is unsolved, and I suspect it will take a lot of time to solve it. So I think I'm running out of time, and in the spirit of this open problem, I want to conclude with this photograph. Sorin, this is the granddaughter, as you know, of Stanislav Ulam, whom you brought to the first RECOM conference in Santa Fe almost a quarter century ago. And happy birthday again.
Do we have any questions for Pavel? I see we've got one question.
Hi, Pavel. That was outstanding. This is Andy Clark.
Oh, hi.
Good to see you again. So you presented the uni alignment as though you had a haploid genome. And I'm wondering, what about the sort of phasing of the haplotypes for a diploid individual?
Phasing haplotype if in a diploid individuals in centromere is difficult, but probably not that much difficult that phasing of the rest of the genome, because there are centromeres is arguably probably the most rapidly evolving region of the human genome. There are a lot of mutations that allows us to separate the haplotypes. So it has been already done for many genomes in the Human Pangenome Consortium. Subject to validations, of course, because it's currently ongoing effort.
Right, thank you.
Yeah, I don't see any other questions, so thank you again, Pavel. We will move to another Zoom talk now by Liliana Floria. Is Liliana online? Yeah, so Liliana was part of our informatics research team at Celera some years ago. She came in having already done really important work on how you handle splicing in studying how RNA is produced from DNA. So really an essential feature to annotating the genome, has gone on to do a lot more important work in that area, as well as much more on the applications of that to many problems in functional and biomedical genomics. So really great to see you again, Liliana, and please go ahead.
Thank you, Russell. Very nice to see you from afar. I'm sorry I cannot join you. I have a laryngitis. Happy birthday, Sorin. I cannot find you, I cannot see you, but happy birthday from the bottom of my heart. So I will start sharing the slides. And the slides will talk about a story that's a little bit down the memory lane, and that takes us into the current years a little bit. Okay. All right, we're set. So I'm going to be talking about unraveling the combinatorial design of genes and designing algorithms that are based on splice graphs to reconstruct transcripts from a variety of sequencing technologies. But before I start, I want to tell you a little bit about my relationship with Sorin. So I actually met Sorin virtually a long time ago. I believe it was 1995. I was a first-year graduate student, and Sorin was organizing. He was one of the organizers in Albuquerque of the first, I thought it was RECOM style conference. And I applied for financial aid to travel. And he replied back to me and he wrote in Romanian. We are both Romanian. So I was very surprised. So that's where our relationship started. Many years later, after a few RECOMB conferences and other meetings, in 2000, I was interviewing for a position after graduation, and I received a message, a quintessential message from Sorin, asking me if I was interested in interviewing at Celera. And I interviewed, and the rest of history. So if I want to give you any thoughts from my talk today, these are the take-home points. Basically, the memories and the teachings that I learned from Sorin, and there were many, but those are the ones that I specifically got from him. So first of all, it was the love for mathematical abstraction, that very deep thinking that underlies the practical problems, all those applications that we're writing in bioinformatics. Second, that in order to have a powerful algorithm and the powerful software tools to go with them, we need to have rigorous mathematical models. Then what I learned from him, and it turned out to be extremely valuable, was how to name tools and also the power of branding. I also learned how to appreciate the uniqueness and the excellent qualities and unique qualities of everyone on my team, and how to join together people with complementary skills. And lastly, for those who might know this, I also learned about the power of the MOAT. And that's the mother of all tables. So, so with that, I'm going to tell you about my journey to investigating and modeling alternative splicing in genes using splice graphs, a problem that originated from my work at Celera and that continued after a few years. So I'm going to present that in phases. Phase one was gene and alternative splicing annotation from EST and mRNA sequences, and you might guess where that started. Celera assembled, and we might agree that the most important problem in genomics has been assembling the human genome. That being said, the second most important problem is genome annotation, identifying the gene structure and identifying the number of genes in the genome, because without that annotation, the genome is not very useful. So there have been a number of estimates for what the structure of the gene looks like, starting with Gilbert's supposition that genes are, eukaryotic genes are organized in pieces, and then with a number of approximations or estimates for the number of genes in the human genome. And those numbers went somewhere between 1800, 877 here in the lower right corner, all the way to 6.7 million in 1964. If you're looking here, the advent of the ESTs, and then here at 40,000, at an estimate of 40,000, between 30 and 40,000, it's the annotation of the Celera genome and also of the public genome based primarily on ESTs, mRNAs, and cDNAs. Okay. So the same technology that gave us, or the same technologies that gave us the human genome, which are the ESTs and Sanger sequencing, have also taught us something very important about the genes. And that was the fact that they are overwhelmingly alternatively spliced. And indeed, when looking at the alignments of ESTs on the genome, we could see that they showed blocks, namely exons, that combine in different ways in order to form messages or transcripts of the gene. So that's a property that's called alternative splicing. Alternative splicing was known. However, the extent to which this occurred in the genome was underestimated. The Celera genome annotation told us, gave us an estimation of about 70% of the genes showing alternative splicing, an estimate that now has been raised to about 95% of the genes. Therefore, annotating alternative splicing is an inherent and a very important property of, and must be a very important component of any gene annotation process. Up until that point, programs for identifying or predicting genes along the genome primarily looked at a linear gene model with one set of exons. However, in light of this view of overwhelming alternative splicing, we had to come up with a new model. And the model that we thought of at that point, and apparently there were multiple groups around the world that were thinking the same way at the time, was that we would use a new data structure that was called the splice graph. So the concept of a splice graph is simple but powerful. The gene is represented as the graph. The vertices in the graph are the exons, the arcs in the graph are the introns, and then the alternatively spliced transcripts are represented as the maximal paths in the splice graph from a node that has no incoming edges to a node that has no outgoing edges. And this data structure was the data structure on which we built an algorithm and then a full-fledged annotation pipeline for the Celera rat genome in the informatics research department. And we call that pipeline AIR. That was Sorin's idea. It's very inspired. We are grateful for that. And we are also grateful for all the support that, and encouragement that he showed us, even in the face of so much pushback from the company and from our colleagues in our departments. So what did this look like? I'm gonna show you what it was looking with an example. We would start by mapping the evidence to the genome. And at that point, we had cDNA evidence, ESTs, mRNA, cDNA sequences. From this, we would extract information about the boundaries of the exons and the introns that would connect the exons and organize them in this compact data structure that was a splice graph. The splice graph naturally contains, potentially encodes many more variants than are realistically biologically possible. So the next step was to enumerate the variants, and we came up with this heuristic scheme for prioritizing the variants so that we would select the ones that are the most likely to be represented in the annotation. And you have an example of a scheme here. We actually worked very hard on that scheme and on communicating and receiving feedback from the annotation team. Basically, we had scores for exons that were based on the coverage of the exon with the ESTs, on whether there were gaps, on how many ESTs we need to cover exons. And then we use these exon scores in a more complicated scoring scheme for splice variants. And I'm showing them here. At the end of this, after a couple of years of work, and this was a collective work between informatics research and the genome annotation team, we were able to produce a full-fledged annotation. We applied that to the Celera rat genome to produce to generate the production annotation at that point. And we also applied it to the public genome so we could publish. And indeed, this paper came out in Genome Research in January of 2005, and it was one of the very first methods that were based on the concept of the splice graph. And it turns out that just a couple of years later, our Celera rat genome annotation and the Celera assembly were also uploaded, deposited in GenBank, where they exist at this point. So that was phase number one, ESTs. Phase number two looks at the transition in technology, as you might have believed. So in the 2000s, when we designed the AIR annotation, we had about 9 million human ESTs, about 300 to 800 base pairs in length, mostly from the 5' and 3' and some from 5' UTRs. And they are all mixed across tissues and conditions. They were expensive and very slow to produce. In 2008, with the advent of the deep sequencing and next generation sequencing technologies, we were able to obtain through RNA sequencing tens to hundreds of millions of reads for a single sample. They were low cost, they were very fast to generate, but they were short, which produced problems. So obviously, they gave us opportunity like never before to identify alternative splicing to finally annotate the genes and their splice isoforms in all possible tissues or conditions, the ones that we can survey. However, there were significant challenges because these reads are short. They also have a number of artifacts, an important one of which is that of presenting intronic reads. So we took to the new task and we took on these new challenges. And now as just starting as assistant professor at Johns Hopkins University, I looked into the problem of assembly, splice graph-based assembly for short read sequences. So we do not have the continuity of longer sequences of the type of ESTs and full-length mRNAs. However, we have large quantities that can tell us something about the flow or about the abundance of of the transcripts, which can be used in something like linear programs or estimation maximization algorithms in order to deconvolute the potential transcripts. And that's exactly what we used in our program CLASS. So CLASS stands for constraint-based local assembly of splice variants. And you might recognize here the structure of our AIR annotation pipeline. So in stage one, we were identifying, we were predicting the exons, and we were doing so by splitting the genomic axis into exonic intervals and then resolving the exons in every such exonic interval by using a linear program. And basically using the continuity, the assumed continuity and uniformity of the read coverage within that sub-interval. So once we had the set of exons and we had introns obtained from spliced alignments, we would combine them into the splice graph. We would then enumerate all the splice variants just like before to produce an overset of all the possible transcripts. And then lastly, and this is perhaps the most interesting, we were again selecting a subset of variants based on them that would explain the patterns of read coverage. And we developed two algorithms. We had two papers that were based on two different representations and two different objective functions. And I'm going to go a little bit through that. So we're looking at here at a number of, we are clustering reads that show the same splice pattern into something that we call constraints. So we have three constraints here, C1, C2, and C3. And we have the number of transcripts. We establish a bipartite graph with two sets of vertices. On the one hand, we have the transcripts, and on the other hand, we have the constraints. And then we draw an edge between a transcript and a constraint if the transcript satisfies a constraint. So with that, the problem is basically to identify the minimum sets of transcripts or some set of transcripts that can cover or that can explain all the constraints, so all the read splicing patterns. And we could model this basically as set cover, showing that we can look at the transcript as being the set of constraints that satisfy. And of course, there's a greedy approximation for set cover that's logging the number of elements, and that's what we used. So basically, at every point, select the constraint that select the transcript that maximizes the number of constraints so far. That turned out to not be enough for us because that would produce the minimum number of transcripts, but also would start with the longest transcripts. We wanted to start instead with the most abundant ones. So then we modified the problem by associating the number of reads with the constraint clusters and taking that into account. So that could be formulated as we call it a quantitative set cover. It's also a weighted set cover, but again, we can use an approximation algorithm that is greedy in nature. We implemented that on the splice graph with a dynamic programming algorithm, which I'm not going to go over, but turned out to be very fast and turned out to be very memory efficient. So bottom line, at the end of this phase two, we had two programs, CLASS and class 2 for reconstructing transcript variants from RNA-seq data. So the question was, however, can we do better? Can we do better? So this leads us to phase 3. If we're looking by and large at an RNA-seq experiment, everything that I've shown you so far has been done in a single sample. However, an RNA-seq experiment usually has multiple samples and typically control and test samples. So a normal RNA-seq analysis goes through producing the alignment and annotations in every sample and then merging the produced transfrags or transcript fragments through a process that I call meta-assembly to produce an annotation that's further fed into differential expression and differential splicing analysis. So what happens is that while at the single sample level, we were able to produce very powerful algorithms with sensitivity close to 50% and precision in the 25 to 35% range in terms of reconstructing full-length transcripts, turns out that this step here has been vastly underestimated. So the meta-assembly would actually bring the accuracy, so the sensitivity would grow by about 5%, but the precision would decrease drastically to about 5 to 15%. That is because noisy transcript from the individual annotations would be assembled and would be represented in this final set. So with this in mind, we thought, can we improve upon both the meta assembly and the individual sample assembly at the same time, perhaps by using the gene information across all the samples? And the thought behind it was that by looking at the complementarity among the samples, we would improve the completeness of the set. Whereas looking at the redundancy across the samples would increase the precision. So we did just so in developing our tool SciClass, which was a multi-sample transcript assembly. And I can say that this is the first one that was actually functional. Okay. So what it's doing is basically starting the same variant, but starting from a very accurate set of parts, exons and introns, that has been decided through statistical methods across multiple samples, then building a global sub-exon graph that is shared by all samples. Samples individually have built their transcripts. The full transcripts that is then reorganized, fed back into the sample level annotation, and finally, the meta annotation is obtained through a voting process. So I'm going to keep this very short, and I'm going to show you only one figure. So here we compare the performance of SciClass with the performance of current reference algorithms, and those are the string tie system and the scallop plus the assembler Taco. So you can look at SciClass being in red, the other ones, the other colors. Precision is in continuous line, and in dashed line you can see sensitivity. So what I'm going to point out is that yes, you can see scallop having a little bit more sensitivity. However, the precision for both scallop and Taco and string tie, after about 20 samples, so after about even 10 samples, drops dramatically. On the other hand, SciClass has constant, robust, both precision and sensitivity, the red lines, with a large number of samples. So at the bottom here, you see we use 2 samples, 5 samples, up to 667 samples in the Jovadi's data set. And we can do this at this point with thousands of samples. We published this, that was already 3 or 4 years ago in 2019. And since Soren asked to think about some questions, these are not exactly open questions, but one question that still remains is, what is the best performance that one can obtain with short RNA-seq reads, and what are the conditions? So how would we need to design an experiment, a sequencing run, so that we obtain the best possible assembly? So that's one question, and I'm sure that there's a good mathematical problem in there, and maybe some of my students or some of Soren's students will look into it. And the second question is, and it's a much more practical question, will long reads replace RNA-seq for transcriptomics? And that's over the next 5 years. So I guess I'm going to leave you with that.
So you're muted, Liliana.
Hopefully, I was not muted for the whole presentation.
Just the last 20 seconds.
My slides are beautiful, and my voice is not very good anyway. So I want to acknowledge the contribution, the friendship of my colleagues at Celera and Applied Biosystems. We have had a great time, and I learned so much during the development of AIR. And then I want to clearly acknowledge Lee Song, my student, extraordinarily talented student, now an assistant professor at Dartmouth, who developed the Class 2 and the SciClass software. So thank you so much, and I miss everyone.
Thank you, Liliana. Do we have any questions?
First off, this is Craig Venter. Thank you for your contributions to the annotation. And one of the problems since our very first EST sequences in 1989 and has plagued all the RNA approaches is because of RNA processing in the cells and the methods most people use for making cDNA libraries, there were unprocessed RNAs containing introns. And so that plagued, obviously, your analysis and others. Can you back calculate the percent of all the unspliced introns, and do any of them have biological significance?
So I would say, so we only observed, so thank you for pointing that out. That was one of the realizations as the new sequencing technology came along, that now we could actually see when we had massive amounts of RNA-seq reads, we could actually see the intronic reads from unprocessed RNA. So now we actually have tools, and specifically the tools that we developed were to identify the most likely cases where this could be eliminated by just looking at the distribution genome, looking across the samples. So we devised a number of algorithms. So looking at ESTs right now, so just based on ESTs, we're probably not able to do that. But I would say that based on RNA-seq data, we now have a much better idea about what constitutes intronic RNA noise and what constitutes alternative splicing of the type of intron retention. So we can't do it with ESTs, but we can do it with RNA-seq, I think, with some level of confidence. It's still a difficult problem, though.
Is that fine?
Hi, I'm Bonnie. I don't have a biology comment. I have an algorithms one. So if you're ever looking, it sounds like you've got set cover and weighted set cover working quickly enough for you. But in 1989 in Fox, Peter Shore, who's here, and John Rumpel and I have a randomized algorithm for set cover. That's truly lightning fast. And in fact, Barish Eckham used it in a recon paper for universal hashing, universal hash functions, designing universal hash functions with Yaron. Yeah. Anyway.
Thank you so much.
Yeah,
Thank you
yeah,
so much
it
for the
is,
reference.
and
Yes.
it uses only pairwise independence. So if you have parallel computing, it's really, really fast. Anyway,
Yes.
and it gets the
yes,
same
I give you-
guarantees as the greedy algorithm. It's provably gets the same
Oh.
guarantees. Anyway, I just figured that might help.
Yes, that absolutely helps, and I really should go back and basically practicing what I said on slide 1, which is to look for smart and for rigorous mathematical modeling before approaching a practical problem.
Thank you again, Liliana.
Thank you very much.
So we're now moving back to the beginning of this session, Gene Myers' talk, which was originally earlier in the schedule. Unfortunately, because of the change in schedule, Vineet Bhafna was not able to be here to give the introduction he had planned, but he asked me to give it for him. So what I will be reading now are Vineet's words, not mine, so please thank him for the kind words. So Vineet writes, Gene's contributions to computational biology are many and profound. In 1990, he published BLAST, which made search of molecular databases tractable. It introduced many innovative ideas, seed and extend paradigm for trade-off between sensitivity and specificity, banded dynamic programming, and others. BLAST remains the prototypical bioinformatics tool with 110,000 citations. In 1993, he took a clever but memory-intensive data structure, suffix trees, and simplified it into a novel data structure called suffix arrays that enabled string queries with large genomic sequences. In 1997, he proposed the use of a whole genome shotgun for sequencing and assembling the human genome. The idea, as Craig reminded us yesterday, was hugely contentious, and the establishment only published the paper if a rebuttal was published in the same issue. In 2001, Gene and his team defended their idea by assembling Drosophila and human genomes, setting the stage for the genomic revolution. Gene continues to be a nuts and bolts scientist, often programming while at conferences, and continues to develop tools that people are delighted to use. He's won a long list of awards, which I will not go into today except for one. Gene was awarded the Paris Kanellakis Award, which is given to one computer scientist for, quote, specific theoretical accomplishments that have had a significant and demonstrable effect on the practice of computing. Please welcome Gene Myers.
Can everybody hear me? Yeah. So it's been a pleasure to be here this weekend and be with all the people that I've walked the road of this life with a piece. You know, it's a privilege and an honor to know all of you. And specifically, happy birthday, Soren. You know, as has already been mentioned, I mean, Soren and I walked the road quite closely together from 2000 to 2002, when we basically co-directed the informatics research department at Celera. As other people have noted, you know, Soren was very productive in that period, finalizing his NP completeness proof of the 3D icing model. And I think the kind of the highlight for me was the first work on phasing and phasing blocks. I remember that very clearly. That was very exciting work. And really he started that whole arena. And another thing that was less known, but actually it's like Pavel and I always give the same talk. So yeah, Pavel stole half my thunder. But anyway, it's, you know, it was the fact that you did the comparisons of our genome to the other genomes, and you did some of the first genome scale alignments. And in fact, hopefully if I get to it, my talk will cover that specific topic, which is still quite germane and active. And just on a personal note, I mean, I'd been at Celera since 1998, and when when Soren arrived, I mean, at that point, I was quite, after 2 years of intense work, I was quite crispy, to say the least, and and it was just, you know, it was like I was so happy when Soren arrived. I mean, you know, other people have talked about how ebullient and enthusiastic he is, and you know, for me at that point, man, I needed that. So thank you. A debt of gratitude. And really it was Soren who, you know, we were basically an assembly team, I think, up until that point. We were starting to branch out in other things like mass spec, but it was when Soren came that really, it was Soren's vision to turn us into kind of a first, you know, world-class informatics group, and he did a lot of the hiring. And, you know, by the end of it, we were at 40 people, and I think at the apex, we probably were one of the best informatics groups in the world, and that was Soren's achievement. Okay, so let me try to give a talk. I was going to do something quite geeky, but I thought just, you know, yesterday I was thinking about it. I thought I'd do a couple of kind of warm-up things that might be fun. And so one was I want to talk about the conceptual effect of read accuracy on assembly. I mean, as you know, the technology's changed a lot over the last, you know, 20 years to the point now where we have, you know, 20,000 base pair reads that are 0.01% accurate. And so what's the impact of that? Okay, what does that mean? And so the original string graph theory, it can be kind of explained as follows. Think of that line as being a genome sequence. Think of the colored arrows as being repetitive sequences. So for example, there are 3 copies of the green repeat. And let's imagine that they're exactly the same. Let's make it as hard as possible. Okay, and the rest of the stuff that's still yellow and thin, that's relatively unique DNA. Okay, and the way to think about it is, I'm not going to give the algorithm, but what the algorithm does effectively, if you thought of the string as being floppy, let it be loose, and you just hybridized all of the repeats to each other. So all the green guys come together, all the purple guys come together, and all the orange guys come together, you get that. Okay, and that's effectively what kind of our quartile collapsing algorithm did. I mean, you know, blah, blah, blah. But in essence, that's it. And in some sense, it took me a long time to realize that this is if the repeats are bigger than the reads, then this is as good as you can go. Okay. The only other kind of augment is if you kind of look statistically at the coverage of the reads on each of the parts of this graph. Right, and as Craig pointed out, the sampling has been relatively Poisson. Okay, then you can actually estimate that the yellow regions are 1x. that they're unique DNA. So you know that the yellow is all one. And in an ideal world, if you've got a perfect collapsing of the repeats, then you would, for example, know that the green guy is a threefold copy because there's 3 unique things coming in and 3 unique things going out. So in any generalized Eulerian tour, you got to go through that guy 3 times. And in fact, you can even, and now this is a little bit special, but if you try to think about how many ways there are to go from the beginning here, from this end to the other end, okay, there's actually only 2 tours. So there's 2 linearizations. Okay, so that's kind of, that's 10 years of theory. Okay, but what's the reality? Okay, so there's one of those collapsed repeats, right? And in an ideal world, I would have loved it if the repeats had actually done that, but they don't do that. What you end up is you end up getting this hairball. Okay, and I'm going to explain why you get the hairball in a minute, but you get this real kind of knotted region where, you know, things are aligning and then they're not aligning, and it's just a complete mess, right? No way. And so what we were doing is we were just thinking of it as kind of a black box problem. I had a bunch of things coming into this junction. I don't know what's going on in the junction. I got a bunch of things going out. And the problem is can I connect those things together? At a minimum, we could scaffold them. In other words, we could tell this input went with that output, you know, using the paired end read, but oftentimes we couldn't get through those regions. Okay, so how do you get through? So, but what's the reality of string graph? I mean, why does that hairball occur? Okay, and so, you know, with the PacBio reads, the initial PacBio reads, the error rate was 10%. Okay, now you're trying to find overlaps between reads at 10%. Well, the 2 reads are going to align at about 20%, right? 20% minus 1%, about 19%. Okay. So, and then, you know, not all the reads are exactly 10%. Some areas are a little noisier, some are less. So you gotta have a little bit of cushion, right? So, you know, in a case of like a 10% error rate of the reads, I would probably look for overlaps that are statistically significant at, say, 30%, okay? So I'm giving myself 10% overhead above the average because there's gonna be fluctuation in the error, right? Okay, so you gotta allow a little more. But if you think about it then, if you think about it, if I have a region where the reads are pretty good, and there's a part of the genome that's repetitive at 10%, okay, then what's going to happen is that thing's going to overlap, but parts of it are going to overlap, but then they're not going to overlap, and it's going to be going in and out of overlap. So so if you have kind of these repeats are not exact, say they're like 10% different, then what's going to happen is you're going to see this kind of fractured pattern where parts of them align, parts of it don't align, and you get a hairball. What happens if you have a 0.2% error rate, okay, which is what's happening with HiFi? With HiFi, so then kind of by the same analogy, if I'm looking for overlaps, I need to look for overlaps at 0.6%, right? That's pretty tight, which means that the only thing that's going to look repetitive are things that are less than 0.2% apart, right? So if I have 2 kind of transposons and they're 10% apart, they're going to completely separate in this model. They are not going to overlap. Right. So basically, almost everything comes apart, right? So you don't get anything that's nearly as tangled. In fact, it's very clean. Some of them actually get resolved, and maybe there's still a few knots, okay? And in fact, if you take the HiFi data, we know that 80% of the errors are homopolymers. So if we compress the homopolymers, then our error rate's down to 0.04%. because we've gotten rid of 80% of the error. So the average error is 0.04%. And if we do a little bit of basically simple consensus across all of the reads, we can get that down to 0.01% error. And at 0.01% error, nothing in a genome is generally repetitive. Nothing looks repetitive, right? So the idea is how repetitive a genome is depends upon the lens that you're looking at, right? I mean, how much of an error lens do you need to look at it in order to accommodate for the technology, the error of the underlying technology, right? And so the more and more accurate it gets, I mean, the more and more clean it gets. And I mean, the thing that I've, basically when you look at most genomes, okay, there are very few repetitive regions that are, identical at 0.01%. There are a few, but very few, okay? So, and the other thing about the technology is the length of the read. So in other words, when you look at this black box model, if the gap is small, then, you're probabilistically going to have a read that goes from one end to the other, and that solves it for you. So the longer the reads also, right? Even the things that are perfect are going to get solved just by the spanning reads. So long reads, 20 KB reads also help, okay? And for large ones, you know, the idea is that, which nobody I have seen ever execute on, you know, in an efficient way, so this is an unsolved problem, is you should potentially be able to use any microheads that are possible to further resolve this. And nobody who's worked on the assembly problem seems to get around to this. So for example, here's the CHM13 from the 2022 publication where they went telomere to telomere. And this is the string graph that they got. And you can see it's very clean, right? It's really, I mean, it's basically, there's a chromosome, it's practically aligned. There's a few knotted regions, right? So, you know, they're doing it at 0.01%. Okay, a little bit of it's still got a few knots, but it's very clean. I mean, there is this rather nasty tangle involving the agarocentric regions where several chromosomes actually intertwine. You can actually even see the mitochondria there, but it's very, very clean. And, you know, what you can observe about that is if you look at the string graph, instead of just kind of the linearization that most assemblers point out, there are no gaps, really. It's like, I mean, this thing connects to this thing. It's just that there's this knot in the way. And so most assemblers will just report that, and they'll report that and say, hey, I don't know about that. Okay, but in the string graph itself, it's still coherent. There's still coverage. There's still stuff there. There's this path. So, and the other one is, as I just pointed out, there aren't many knots. So if you take one of these, for example, like this one here, and expand it, I mean, here it is. And so what are you doing? I mean, it's actually the problem that I initially gave you, the beautiful problem, which is I want to find a tour through here, right, that explains all the data. I want to come in here, walk through this thing and walk out, okay? And the problem is, how do I do that? Well, you know, I can't help but wonder if you couldn't do it by looking for heterogeneities, you know? What they did was they basically produced lots and lots of ultra long nanopore reads, over 100 KB, and then used the nanopore reads to basically thread their way through to discover what the path was. And they resolved most of the path that way. They basically also used some elbow grease. So it's brute force and not diploid. Yeah, so I think that, you know, we can do telomere to telomere genomes. We can do them from end to end. It's still somewhat heroic in terms of effort and expensive because of the amount of, you know, like long reads you need in order to thread the ambiguous regions, okay? But we're getting very, very close. And like I said, I think that, you know, but to me, the one thing that's disappointing is that really the algorithms haven't changed much. I mean, they've changed in response to the scale and the nature of the data, but the overall algorithmic paradigm is not changed, okay? It's just, it's unfortunately, it's a fail for us. The data has just gotten good, okay? So there's one last chance, okay, which is, can you thread some of these guys by looking for the microheads, okay, by actually looking at the microheads, all right? Okay. All right, then I want to kind of motivate why I think that genome alignment now is more germane than ever. And then I'll kind of get to the technical meat. So, I mean, what's happened now that we can produce these beautiful telomere genomes is that there's a lot of projects now underway to go out there and sequence nature, you know, basically find, you know, basically catalog all of the creatures that are out there and everything that we can get, you know, 10 to 100 nanograms of DNA from an individual. We can sequence and produce one of these beautiful genomes. And we can go telomere to telomere. There might be a few gaps. I mean, we don't worry about the gaps so much, but we can go telomere to telomere. And of course, it would be nice if we could actually fill the gaps by being able to understand the tours. So, and this has all come about because these long reads mean that we can do nearly complete genomes. So now that we actually can really produce genomes that are worth cataloging, you know, putting in at least people feel that the level is such that now we can, you know, we can make a catalog. We can make, you know, we can do Linné again, okay? Except now, you know, we're going to go through and sequence everything. And so there's a lot of projects. I mean, the first one, you know, started at Dulles Airport in 2016. It was Eric Jarvis, Richard Durbin, Adam Philippi, and myself basically getting together saying, let's get going. Let's see if we can do at least 260 orders of the vertebrates. And so we started the Vertebrate Genome Project. And at Dresden, we did 20 of the species for the 260. We also, at the same time, I met a enchanting, charming bat biologist by the name of Emma Teeling. And together with Emma and her associate, Sonia Vernez, we basically started a group to really focus on the 1,400 bat species, because bats are extremophiles. Bats live longer, way longer than they should. They never get sick. They eat sugar all day and don't get diabetes. They're pretty cool. They're the extremophiles of mammals. And so I wanted to do those guys. And it's at Dresden, before I left last year, we had done 42 species for a total of 84 gigabytes of, high quality genome. And then of course, why not do everything, right? So in 2018, Harrison Leuven basically said, okay, and basically the Earth Biogenome Project is actually a coordination project, which is trying to coordinate all of these efforts. So, and I'm not going to do all of them. There's a group that's out there trying to do insects. There's, there are groups doing all kinds of collections. But I think I want to point one of the ones that I think is interesting, which is in 2018, the Sanger Center decided to start the Darwin Tree of Life project, where the focus is to do actually I say 65, we know there are now over 70,000 species of life on the British Isles, okay? And the reason I'm pointing at that one is because instead of it being about a clade of life, it's about an area, about an ecolocation, okay, which I thought was interesting. And the other thing that was really smart about it is that, there's this Nagoya Protocol, which makes it very difficult to get samples sometimes. For example, you can't get a sample out of Ecuador because there's nobody to talk to, but they've signed the Nagoya Protocol, so you can't take anything out unless you talk to somebody in Ecuador, but they don't have a department to talk to. So how the hell do you get something out of Ecuador? You can't. Okay, there's a lot of problems like that, okay? So we can't get these things. So we can't get a lot of things, but in this project, notice it's their country, okay? It's their kingdom. No problem. And the other smart thing was that, there's a long history of cataloging everything that lives on the British Isles. So they already have exquisite, beautiful catalogs for most of it. Although it's interesting, once they start looking, they're finding things. Kind of not at the level that Craig was finding them on Sorcerer 2, but there's still things popping up. And then the Europeans actually joined the same idea. And just, kind of to brag a little bit, at Celera, we've done basically over 160 genomes totaling 200 gigabytes of basically beautiful completed sequence. And here's our list, okay? And so I have these genomes, and notice the quality of these genomes. The ones that were done with HiFi, the Contig 50 is over 10 meg, okay? And the Scaffold N50 are the chromosomes. They're telomere. And, I've got these on my laptop, and I want to compare them, okay? And, how can I do that? And I looked at the literature, and not too long ago, Eric and a couple other people basically wrote a paper about aligning 600 vertebrates with, I don't know, whatever exists out there, cactus. And it took 1.9 million CPU hours. Okay. And I looked at that and went, no, it's scalable, baby. Okay. Because we're going to have thousands of these things soon. Okay. I mean, we're going to have thousands of these things. It's, we're just kind of getting going, okay? And in the next decade, you're going to have 10,000 high-quality genomes. They're basically covering, all the vertebrates and the clades. And I mean, we want to understand what their evolutionary relationship is. And these are not like, Pavel was talking about human to human, where, really I consider that to be a close alignment. We're talking about things that are diverged by 50 million years. So you expect, 30%, 40% alignment. Okay, so I'm going to talk about sorting for speed then. So how am I doing on time? I don't want to go over. Okay, I'm already at 18 minutes. I got 12 minutes. Okay, well, I'm not sure. So the main point that I want to make with this slide is that cache coherence is a big thing, okay? So you want to get something from the memory. If it's not in one of these series of caches, which I've shown in this complex diagram, it's not in the L1 cache, it's right next to the CPU, then you're going to suffer a factor of 50 when you go to get it, okay? And people don't tend to think about that, okay? But that's huge, right? So if you have a program that has a random access pattern in the memory, it's not going to be very fast because the caches aren't going to be able to figure it out, right? They're not going to figure out what you're doing. So, you want to try to, the short of it is that because of this cache hierarchy and the discordance in speed between what the CPU can do and what your big, 64 gigabyte memory can do. And this is the structure for this laptop, by the way, is you want to arrange your computations in a small number of simultaneous sweeps. If you go through memory in a, you know, in a sweep in a pattern that the hardware can predict, and it's also prefetching, it's going to go fast, okay? Because the prefetching and the fact that you're pulling blocks at a time, okay? So, so the general idea is as follows, and it's a general paradigm, and I've used it for a bunch of problems, and I probably shouldn't give it away because you guys will be able to compete with me, okay? But I'm going to give it away, okay? Because I love y'all. Okay, so, well, you know, and I'm getting old. So you want to find, like, say I'm going to take a problem. You want to find seed Kamer hits between two corpuses of data, G1 and G2. Maybe I call them G for genome. And so the way you would typically do this is you build some kind of index of, say, the second genome, okay? So you build a suffix tree or suffix array or an FM index or, you know, whatever, okay? And then what you would do is this, our hash table. And then what you do is for each position of G1, you would look at the string in G1 and you'd look it up in S and see if there's a match, right? That's what you would do. Okay, that's the way most people write that code, right? But that's not cache coherent, okay? I mean, you know, each consecutive string as you're going along G1, I mean, they're completely different, don't have anything to do. You're going to a completely different place in your index, right? It's cache, almost a guaranteed cache miss every time, okay? So you're running at about 1/50th of what you could be running, okay, with that step. So here's the simple thing. It's kind of counterintuitive. What I'm going to do is I'm going to build a list of strings of Kamers and their positions in pairs. I'm going to bundle up the Kamer with its position. And I'm going to build a big list of those for G1, and I'm going to sort it on the Kamer. So now I have a sorted list of the Kamers, and for each of the Kamer, I know where it came from. And I'm going to do the same thing for G2. Then what I'm going to do is I'm going to merge G1 and G2, right? Looking for the Kamers that they share, right? And it's a merge, so it's sequential, which means it's cache coherent, right? Because I'm sweeping, okay? And then what I'm going to do is I'm going to build a list of the pairs of hits, P1, P2. At P1 and P2, there's a common Kamer, and then I just sort them on P1. So instead of kind of doing the lookup, I'm going to do 3 sorts. That looks like it might take longer, right? But if the sort is cache coherent, and it's a good sort, it's a really rockin' sort, okay, it's going to win. And it's going to win by a lot, okay? And that's the secret sauce. And you can solve a lot of problems that way. I mean, if you think about it, what I'm doing is I'm taking all those queries in G1 and I'm packaging them up and I'm sorting them in a good order to look them up, right? So the way is instead of asking each question individually, I'm taking all the questions, packaging them, sorting them, and getting them ready so that I can answer them in a big sweep, right? Because that's the way a lot of, I mean, if you only want to look up one string, then I would agree, just build an index, okay? But if you want to, if you're doing, you know, you're comparing 2 large corpuses of data, sort the queries, okay? And then sort back, okay? So there's a thing called the MSD radix sort. I don't want to, I'm already in danger of running out of time, aren't I? Yeah, I got 7 minutes, okay. So there's a thing, it's the most significant digit radix sort. It's a really cool sort. People have been talking about it for about 10 years. You can find a lot of literature about it. I mean, what you can do with it is basically, if you have a set of n strings, so a huge number of strings, like, you know, 1 billion strings, okay, n is 1 billion. And there are strings of some length k. Let's say k is like 40. We're doing 40 mers. Over an alphabet sigma, let's say sigma is DNA, sigma is 4. Okay, it will do this, and it's a cache coherent sort. Normally, if I have more time, I actually kind of do an animation of the MSD sort and show you why it's cache coherent, but I don't have time for that today. I'm just going to cut to the chase, which is so you can build this highly optimized MSD sort, which is cache coherent, and it takes, I don't know why the time didn't come up. Sorry, I blew the animation. It takes order n k squared time, okay? it takes actually, it does order NK comparisons. In other words, it does a linear number of comparisons, Mike, but it has to move the data and the data of size order K each time. So it's order NK squared. And you can sort anything. So if you wanted to, example, sort 64-bit integers, you just view the 64-bit integers as 8-digit radix 256 numbers, right? Or in other words, you view them as 8-letter strings over a 256 symbol alphabet, and you do the sort. So you can sort integers, you can sort strings, sort everything. It's completely general. And you can produce records, and you can just sort the records. So for example, oftentimes I'm producing contig position kind of pairs. So in other words, just not a raw position, but the contig that it came in, the position of the contig, and then the Kamer. And I can sort those by the contig position, or I can sort them by the Kamer. I can do whatever I want, and I just carry the data along. The records get sorted. The only caveat is if the record is of size W, then the sort, instead of being NK squared, is NKW. But it's still amazingly fast because it's cache coherent. Everything is cache coherent. And you can even do memory mapping. So this thing could be a file, and you just do memory mapping, and it works gaga. I mean, it's really amazing. You can have some, 50 gigabyte file out there, okay? And you can memory map, and it will sort that 50 gigabyte file like you wouldn't believe, okay? Although I tend to choose things so that they're 12, there's the sweeps, okay? So, so I've used this trick for several things. The first thing I used it for is a Kamer counter called FastK, which does a lot of cool things. And I think I'm just gonna skip over that. And I've used it, I'm currently using it to build a genome comparator, which is what I wanna at least spend just a few minutes on. And then, I've got a very nice scheme for doing something like Bonnie was doing, where it's an Abe-Ruin graph based on some different concepts, but it's going to have the same underlying idea. And I think it'll probably be in the same ballpark as what you've been doing. It'll be interesting to see what happens in the details. Okay, so I'm going to just blow through FastK. It's got some unusual features. And one of the cool things about FastK is I've actually written all of the major applications for Kamer counters, in other words, GenomeScope, SmudgePlot, CADTools, Mercury, and Murfin, in a way that uses FastK. So you don't have to download and mount, 3 different Kamer counters, just FastK will do them all, okay? So that was a big engineering effort on my part. That was my morning coding exercise for like 9 months, I think. Okay, and then, so I want to talk about whole genome comparator real quickly. So, the basic genome aligner is kind of the blast idea. You find a seed, and then you extend, right? You find similar regions. You want to try to find regions where you think there's some basis for similarity, and then you want to try to extend that into a proper alignment. So there are a lot of ideas for seeds. I mean, matching Kamers. The problem, as kind of Pavel pointed out, is it's hard to pick K. If I pick K too small, then I get a lot of false positives, and I end up spending a lot of time looking for alignments in regions where there's nothing. If I pick K too big, then I lose sensitivity. I'm not going to find regions where I should be finding things, okay? Then Pavel talked about mums, okay, which is an old idea, actually. It's like a 15-year-old idea. Maximal matches, I think it was Mummer, it was Art Delcher and those guys, right? So, but I think uniqueness was a little bit specific. The idea that I liked was the idea of an adaptive seed, which is a slight relaxation of a maximum unique match, which is that you basically are adjusting length so specific for the region. Specifically, an adaptive seed at a position P of a genome, so at some position, is the longest string beginning at P that is also somewhere in the other genome. Now, that doesn't mean that it's necessarily unique. It might occur 3 places in the other genome, or that particular sequence may also be twice in the genome you're talking about, right? But the point is it's the longest substring that's specific at that location for that location. So it's called an adapted seed. But sometimes, the seed can occur too many times. And so there's a user definable threshold F that decides when a seed is repetitive. And I usually pick F to be 10. Okay, and so seeds that occur the more times than that, I let them go. So the last idea, this is really annoying. I've got the timer on, but it keeps going blank. So the idea was to build a suffix tree the way it came out in 2011. This is an old idea, but a really great algorithm, actually. I think a lot of people don't use LAST. I think it's under valued, builds a suffix tree or array of S, and I think that's really kind of slow. And then for each of the positions of G, it determines the AC by traversing and finding the longest things, kind of the same way that Mummer does. But as I pointed out, that's completely not cache coherent. So what I do is I build a simple genome index. I don't even build a suffix array. All I do is I build a list of all the 40mers with their positions, contig position pairs, and their orientation, and I sort them. So my index is a sorted list of the k-mers. Here it is over here, okay? And so here are the k-mers. Here are the first ones, 38 A's followed by CG, 37 A's. And, basically these are the positions at which this string occurs in the genome. And I give the contig and the position and also the orientation. So it's canonical k-mers. And the kind of cool thing is that I also, as Pavel mentioned, I use the longest common prefix of this guy with a guy preceding it, okay? And I keep that. So in other words, for the next entry, I know that it's LCP with the entry before it. And that you get that for free out of the sort. I do that with an MSD sort, of course. If you think about it, and I do it for 40mers, it's a poor man's suffix array, right? It's a suffix array that I truncated at 40, okay? It's a poor man's suffix array, okay? It's like, cheap suffix array, because honestly, if I have a seed that's 40 base pairs long, I'm happy. I don't need a longer one, okay? But if I'm down at 19 and it's the difference between 19 and 22, then I want to do the difference. And in fact, typically, I showed you the beginning of the list. If you kind of get further into the list, you'll see that most strings are unique and occur once. Okay, so most of the entries in it, it's fairly big, but it basically takes 30 seconds for each gigabyte of genome. So human genome, 90 seconds, my bats in a minute. Okay, so like I said, the search thing isn't cache coherent. So just very quickly, what I do is instead I have I have an index for G1, I have an index for G2. They're sorted lists of k-mers. I'm basically going to do a merge, right? But I'm doing a merge where I'm not looking for the exact same 40mer, I'm actually looking for the longest common prefix of this one with something in the other one, okay? And I'm not gonna try to explain that picture too complicated, it'll take too long, but I basically can do it with 3 fingers, and using the LCPs, I can get it down to 3 character comparisons per lookup, okay? So it's blazingly fast, right? I'm basically I just scan through that thing, and I can pop out all the pairs of contig positions, right, where there's an adaptive match along with the length of that seed, right? And then just to kind of again, too much detail, I don't quite do it by position. I actually do it in a way in which I get the bands. I get bands of width 64 or any band size that I want, really, but I like 64. Seems about right. And so if I think about it, if I do the MSD sort on the contigs and then the positions, right, then I've got everything lined up to look for chains of hits, right? So I do another sort, okay, on contig position pair to get me to, so that I can easily see a chain. So I see, all the seed hits that are in a given band and between a given pair of contigs are going to be coherent in the list. And in fact, they're going to be in order of their position along the first genome. So I can just very quickly find them. And the problem is that the hit may cross a band. So I actually look at pairs of bands. Okay, that way I get them all. And I do a little thing where I make sure that the projections back to the genome are significant, because sometimes if you have tandems, you get this kind of thing where you don't get much of a projection. That's all parameterizable. You can change those parameters if you don't like them. And then I use a very rapid version of my wave algorithm, which some of you guys know, okay, to basically then extend those into alignments. And so I took 2 bats. I ran this morning. It took 2 bats at 2 gig. It took 3 minutes and 50 seconds to find the alignments. It basically aligned 50% of this bat to that bat. 50% of the thing was alignment. And these are not like, 1% alignments. These are like 25% difference between the two things. It goes down to 30%. So I found 639,000 non-redundant alignment pairs of length, on average length 1,800. Some were longer, some were more accurate, but it does the whole thing. So basically it's very sensitive and it finds everything. And the thing I don't have time to show you is that basically, when I look at the outputs, you see beautiful syntenic runs. I mean, you see the syntenic blocks between the genomes. They're there. And so what I'm working on now is that what this tool does, if you think about it, is it produces the alignments. It just produces anything that's similar to anything else. Could be a repeat, whatever, and you see the repeats. But what you really want, right, in a genome comparator is you want to find the alignments that are due to homology, due to the fact that this region and this region evolved from the same region of the genome, right? So homology is different. And for that, you're basically looking for long syntenic runs. And so I'm working on that part of it now. But this is just an aligner. I consider the other problem to actually be finding the homology, of finding the synteny to be a different problem. And the thing is turning out to be much more versatile than I thought. For example, I took two haplotypes of a butterfly, and I basically compared to each other, and it basically lined up the haplotypes. So it turns out it's a great haplotype alignment tool. Okay, it'll find the haplotype alignments. It'll tell you which parts of your face. Because the problem is you run the assembler, you get phase separated chromosomes, but you don't know what the correspondence is, right? When it comes out of the thing. And this is like actually a persistent problem. So I'm using it for that. Okay. So yeah, I'm just going to thank the people that gave me input and I worked with and helped me, and that's it. So I was almost on time.
Do you have any questions, Eugene?
Uh-oh.
And this is more a general comment for everybody working on assemblers. What I seem to find missing in bioinformatics is bio. And what we want to get, comparing human haplotypes or human genomes, is medically relevant information if we're trying to protect that information versus telomere to telomere genomes, which are nice and sexy, but don't necessarily tell us anything. We want to know with the two phased haplotypes, what leads to heterozygosity. Is it insertions, deletions?
Yep.
Is one haplotype totally inactivated? Is one expressing everything? Are they actually both expressing two different structural copies of an enzyme that are both biologically active, but with different biological activities or kinetics? So maybe the comparison problem is simpler in the sense of maybe concatenating the genes, or eventually, if we can understand promoters, the promoters. It needs to be a biologically driven question, at least from my view as a biologist or somebody trying to use genomes to give medical advice to somebody.
Yeah, I agree
So
with you
in a
100%.
way, you're trying to solve bigger problems without solving the fundamental problems that we need solved.
So thank you, Craig. I mean, I agree with you 100%. But it's the, I view that what I've done so far as a foundation.
It's not bad.
No, it's a foundation. It's the foundation, right? I mean, it's like you need to basically understand what the set of alignments are between the two genomes, and that has been taking an inordinate amount of time. I mean, you may not have been impressed by the 350, but I don't know. I mean, was anybody impressed? I thought it was pretty good speed. Okay, most of these things take overnight.
Yeah.
Today they take overnight when you do one of these things, and so. So the idea is you start with the alignments. Like I said, I don't consider this to be a finished piece. Now, I'm not interested in a pangenome. I'm interested in kind of evolution of things that are 30 million years diverged. So I want to understand kind of what is homologous in terms of evolution and synteny. And I view that as, so now that I have the alignments, I'm now working on codes that are analyzing the alignments, trying to understand what the long syntenic runs are and what the relationships are, where are the homologs and the paralogs. And I would do the same thing. And if I was doing a pangenome, I mean, I could use this thing to compare two human genomes. And I would write a program, a second program behind this that would now look at the alignments to basically look for the biologically relevant features that you're talking about. So this is a work in progress. And I agree 100% with everything you say. And I hope
So, yeah, I'd like to make a comment and to point out to Craig that yours is not the only position to stand on. There are biologists, there are mathematicians, there are computer scientists. And I'll quote the great man, Stan Ulam, ask not what mathematics can do for biology, ask what biology can do for mathematics.
It's true. I mean, and the things that, so Craig, what you've done is you basically gave me my to-do list, okay? I mean, it's great. It's the to-do list,
Mike,
right?
I have the answer to your question. The biologist can give mathematics a reason to exist.
All right, now let's keep it friendly. Let's keep it friendly. But Craig, in a way, I mean, what Mike is saying is that I listened very carefully what you said, because that's the to-do list for pangenomics. But I provided a tool that will allow you to do pangenomics at a rate that's reasonable. I mean, I provide, I'm always the guy in the boiler room, more speed, Scotty. Okay, that's me. Okay, so I've got more speed. Now I need to give you some biology. I agree.
But isn't my problem a simpler one to solve?
No, not really. It's like, for example, when I'm looking for syntenic runs, 90% of it is obvious, 95% of it's obvious what's in a run, and I'm going to spend 80% of my time thinking about the other 5%. I know it.
I think I have a simple problem. It's probably just my misunderstanding, but if you have 100 copies of a TE spread all over the genome, why does that not botch your cache coherent trick?
If I have a what?
100 copies of a nearly identical TE, you're going to have this multi-mapping problem. So why doesn't that botch the cache coherent trick?
I'm not sure. I mean, the T will be sorted to a single entry, and actually that will be considered repetitive. It won't even occur in the index, because I cut off anything that occurs more than 10 times.
Oh.
So this thing will not align centromeres or telomeres. Okay, I'm not solving the same problem as Pavel.
Sorry, one more question.
Oh, okay, all right, sure.
You have time. Have you thought at all about applying this to searching 16S databases and other conserved sequence databases, where your basic k-mer seeding approach is just going to seed to 10,000 different hits that you don't?
Yeah,
really
no,
want?
I would have to think about that. I would do something else. I mean, I would basically try to take the 16S database and compress it and produce a compressed representation, I think. Yeah, no, it won't solve that problem. Okay, thanks, everybody.
Okay, so next up will be Giuseppe Lancia. So Giuseppe came to our field from a background in operations research, really an expert on combinatorial algorithms, who did a lot of fantastic work on algorithms for computational biology, including with Celera. And he tells me he's going to talk about things that have nothing to do with biology today. So we're looking forward to hearing about that.
Okay, thank you. Okay, so I thought at this point you might be tired of genes and sequences, right? So actually, I haven't been working in the field for a while. Where is Soren? I wanted to say something about our meeting. Well, I
went that way, that's all I know.
Okay, well, I can tell something about how we met. and when. But what's that? Okay, I'll do it at the end. Yes. Okay, so the talk is going to be about the traveling salesman problem, which is a very famous. Oops. Presentation mode. Okay, so Soren is back. Okay, Soren, I wanted to remind me and yourself on how and when we met, which was basically 25 years ago now.
Wow.
And it was 98 when I was first I was at the SODA conference in San Francisco. We had a paper with Vineet actually at the time. And then I came to visit Bob Carr, basically a friend of mine that was at Sandia, right? And we met, I gave a short talk, and then you said, do you want to interview for a position like a postdoc? And I said, well, what do you mean by interview? Because I was working in Italy, we don't have these interviews, right? And basically it turned out that it's just going out to dinner with a lot of people, right? And so I'm pretty good at eating, right? So I think it worked very well. And I say, okay, but then there was 1 year of clearance for military, security, because Sandia was like a military base. And then I came, and then you went to Celera, and you asked me, do you want to join? You made me an offer I couldn't refuse. So. And so another year, but then eventually I went back to Italy. But I'm very glad, that our paths crossed, and also I'm very glad that you insisted that I come here, because I wasn't in the States for a while. Okay, that's it. So the problem is the traveling salesman problem, which is a very famous problem that has also applications in computational biology. And basically, you have a complete graph. And distances between the nodes doesn't have to be the geometric distance, right? But any number between every pair of nodes. And you want to find a tour, like a Hamilton cycle, of shortest total length. Okay, so this is a very famous, maybe the most famous combinatorial optimization problem. One way to solve this problem, not exactly, but perhaps with a good solution, is local search. And it's something like this. You start with a random solution, a random tour, and then you find a move. So you change your tour into another tour, which is better than the current tour. Right? You find this move, if there exists one, and then you repeat. So every time you change your tour by applying a move. But if no move is improving, if you don't find any move that improves your current tour, then you stop. That's your local optimal solution, and you return this tour. So in this talk, I'm going to focus about finding the best possible move. So it's not just any improving move, but the best possible move. Okay, what type of moves are we interested in? We are interested in the so-called two-opt moves. So basically, you have your tour, you have your current solution, then you fix two edges of the tour, from I to I plus 1, from j to j plus 1, and you try to remove these edges and replace them with these new edges, ij and I plus 1, j plus 1, so you have another tour. So if the cost of the two edges that you take out is bigger than the cost of the two edges that you put in, then that's a good move, and you do it. And you want to find basically the biggest possible value of this move. So that's your problem. Find the best possible two-opt move. Okay, we call the edges that you take out the pivots of the move. So you have two pivots, and you have to find the best pair. Okay, the value of the move, as I said, is the cost of the edges that you take out minus the cost of the edges that you put in. And this has to be maximum. Okay, so how do people find the best move? Usually, they just do a complete enumeration of all possible pairs of edges, because it's quadratic, it's a fast algorithm, it's considered a fast algorithm. So not much research went into speeding up this simple algorithm, because this is a quadratic algorithm, but we want to make it better than quadratic. at least on average. So what's the idea? The idea is very simple. If you want this expression to be big, to give you a large value, it means that the two edges that you take out must be large, must have a large value. And in particular, at least one of the two has to be large enough. So if you want to move a value, let's say 100, then you have to take out two edges that together are at least 100 of length. So at least one of them has to be at least 50. Okay, so the idea is look not for every possible pair of edges, but one of them has to be large enough. And by large enough, I mean half of the value of the move that you want to beat. So we propose this new algorithm. Basically, it's a greedy algorithm, but it is exact. So you consider all the edges according to their value from the largest to the smallest by using a heap, which is a very good data structure for doing this. And every time you pop the top of the heap, if it is large enough, then you consider all the moves that include that particular edge. And if it is not large enough, then you stop because you have already found the best possible move. Okay, so it's not all pairs, but picking them from a maximum heap. Okay, so we implemented this, and we observed that this thing is subquadratic on average. So here, for instance, you take instances in which every edge of the graph has a random cost in 0, 1. Okay, and here is the size of the instance, the number of nodes, and how many moves you evaluate to find the best one. Okay, originally it was n squared with the original algorithm. Now for this type of uniform instances, we draw this plot and it fits very well with the n to the 3/2 function. Okay, so for uniform instances, we get this average complexity empirically. And for geometric instances, like basically the problem is connect points in the plane, in the unit square, random points in the unit square with the shortest tour. On that kind of instances, we get even better results. So the algorithm is linear. It's fit very well by a line, which is the best possible solution, to find the best move out of n squared possibilities in linear time. You can do better than that on a tour that has n edges. So this is the empirical evidence that the algorithm is good, but we needed a theoretical justification, some proof that this is exactly the case. So the proof seems to be very hard. So we couldn't really manage to give a formal proof that that algorithm is of the expected time complexity, less than quadratic. But we found some indirect way. Indirect or indirect? How do you say? It's the same. Okay. Okay. So what's the indirect way? We introduce another algorithm, a family of algorithms, like a heuristic, in which basically you fix a threshold which depends only on n, and then you look at every edge of the tour that has length at least delta of n, and you consider the moves that include one edge of that type. Okay, so now this is a heuristic, because if delta of n is large, then you do not consider many moves, because only few edges will be large enough. So, but you have a fast algorithm, but it might not find the best move. And if delta of n is small, then you definitely find the best move, but you have maybe to expand all possible edges. So basically, we studied how to tune this delta n so that this kind of heuristic is fast, less than quadratic, basically, but still succeeds with high probability. And then we compare our greedy algorithm to heuristics of this family. So that's the kind of indirect way to prove the complexity. So the main problem, the main theorem basically is something like this, that for every probability, you can find one algorithm of that type that I just mentioned, where delta depends on the probability. And the complexity of the algorithm is actually theta of n to the 3/2 for uniform random instances, or linear actually for Euclidean random instances. So the kind of complexity that we observed empirically. So for these heuristics, and then our algorithm runs faster than this type of algorithm on a random instance with probability at least p. So it's saying, okay, we don't exactly know what's the average complexity of this, but it's dominated by an algorithm that has this kind of complexity. So it's fast. Okay, I think I'm going fast enough. Okay, so with the open problem, besides, losing weight, that's an open problem for a while it's been, and especially after this conference, it's become a bigger problem. Anyway, the open problem with TSP is something like this, is that 3-opt. So if you remove 3 edges instead of 2, then also it's a move that can improve a tour. Okay, and there is a conjecture, not by me, but by some very good people that say that there cannot be a better than cubic algorithm to find the best 3-opt move. Worst case, okay, I was talking about average case, these are worst case, better than cubic, because if there were one, then the same algorithm could solve a lot of problems, such as the all-pair shortest paths, that nobody has ever found a better than cubic algorithm. So it's something like the NP completeness kind of equivalence of complexity. If you solve this, you solve many other problems that nobody has ever found a better than cubic algorithm for. Okay, so basically, I guess this concludes my talk, and thank you.
So our next speaker is Sridhar Hananhali. So Sridhar has really had a remarkable journey through computational biology. He came to Celera best known for some very theoretical work with Pavel Pevsner and Vineet Bafna on how to do rearrangements in genomes and showing surprising result that this was efficiently computable. Switched to be much more biological than theoretical over the years, really getting into the weeds of how genome regulation works. And in the past few years, moving to cancer biology, where he's now head of the cancer gene regulation section in the cancer data science group at the National Cancer Institute. So Sridhar will be telling us, I think, about some of that.
I'll try. Is Soren here? Because our few slides are for him. I cannot even start without him. Okay.
You'll have to rearrange.
I will have to rearrange. That's right. No? Okay. I'll tell you about Soren anyway. So, Soren is not only a leader, but a cheerleader for the community, for younger colleagues and students always. And he is quite a glue, right? Not only across fields, as we see here, but even within the community. And this is one of the anecdotes I picked up. So this is I was doing my postdoc at USC at that time. And me and Vlado and Muthu, we had this paper, something to do with this HP lattice keeps showing up in this conference, some theoretical results. And we submitted the paper, and Soren was one of the editors, and he said, "Oh, there's another paper that was submitted about on the same topic. Why don't you guys team up and write just one paper, one good paper instead of 2 bad papers?" So we did that, and now I have co-authored with some luminaries in the field. Good for me. And then he invited me and Vlado to come to Sandia to spend a couple of weeks and lots of cool brainstorming sessions. Great fun. And then 1997, the first recomp, I remember very fondly. And as you can tell, Soren does not skimp on social aspects of conferences. And that's what I really loved. Remember from that recomp, the social program was really good, not to mention the scientific part. Okay. So then from my postdoc, I went to GlaxoSmithKline, and then in 2000, Soren recruited me, and I was more than happy to oblige because a lot of my friends are already at Celera, and that was becoming a really hub for cool research. So I immediately promptly moved. By that time, Gene's team was already tying the bow on the genome assembly part, and which was fine for me because I was more interested in what's in the genome, not the assembly part itself. And it was ready for me to start doing my work. And there's some of the first papers I wrote when I got there, promoter prediction you mentioned, and there is a story behind it. I won't get into that. And this might be the first real application of what came to be known as phylogenetic footprinting, where you look at multiple genomes and look at the conserved non-coding regions as a way to guide search for regulatory regions. So this was the first application of that. Since then, and this was really beginning of my love affair with the whole gene regulation field, and that's what I still do. Now it's more focused on cancer, but still I'm interested in the nitty-gritty of how genes are regulated. Just to set up the problem, what I'm going to talk about today quickly. So here's the for mathematicians and physicists in the group. This is genome, and here is a gene. Here is the what we call the transcription start site. That's where the gene gets transcribed into mRNA. And here are some regions, what we call enhancers or regulatory regions, that through looping of the DNA make a contact and truly regulate the expression of that gene. Okay. Now, ultimately, we all are working on this genotype-phenotype problem, isn't it? Like, what do these mutations mean? What do polymorphism mean, and so on? And so let's look at the genotype first. So if you see a polymorphism or a mutation in the part of the genome that codes for a protein, then you have some way of pursuing it experimentally. You can look at its potential structure and see where the mutations is, how that could affect interactions, binding pockets, and so on and so forth. But when the mutation or polymorphism is in the non-coding region, that's where we are usually stumped. We don't know what to do with those. Okay. So what we really need is some way of figuring out whether or not a single nucleotide change in the non-coding region could be functional. Okay, so that's the basic problem. And 95% of all the mutations and polymorphisms fall in this non-coding regions, as you would expect by random chance, because coding region only covers 2 to 3% of the genome. All right, so that's the problem. Now, there was the question about machine learning came up a couple of times in this meeting. I'm all for mechanism, but there are times when machine learning is the way to go, right? So this is one of those times for me. So to answer that question, what does a mutation or polymorphism does in a regulatory region? Me and many others for the last 15 years have come up with various types of mechanism-based modeling, okay? But ultimately, it's a very complex, non-linear relationship of binding of various transcription factors that many of them we don't know about that ultimately make a region pick, okay? And we don't know all that. So when we don't know too much about mechanism, but we do know the scope of where we need to look for these signals, for me, that's a really good application of machine learning or deep learning. All right. So this is the general challenges of identifying non-coding, functional mutations or polymorphisms. In the protein coding region, a single mutation can change the amino acid that can make a huge difference in functioning of that protein. But that kind of large effect size is not seen when you have a single nucleotide difference in the non-coding region, right? Effect size is small. And that's partly because multiple regulatory regions together regulate the expression of a gene. So one mutation in one enhancer may have negligible effect on the final outcome, which is the expression of the gene. So I will skip these parts for now. So those are the challenges. So I mentioned machine learning. Let's go with deep learning. So the bottom line is this. We have the input is a DNA sequence, maybe 1 KB long. And I have two questions. One, is this DNA sequence likely to be an enhancer or a regulatory sequence? That's one question. Second question is, can I tell if I change one nucleotide from A to C, let's say, is it sensitive enough for the model for me to say whether that single nucleotide change will make a difference or not in the enhancer-ness of that sequence? Okay. So we implement this deep learning model. In goes this DNA sequence, out comes a number, which is the enhancer regulatory potential of that 1 KB region. Okay. So fine, I mean, we have some nice training set, we train it, now we have to show that it works. That's just the beginning of the project. Okay, so this is content of this paper. Right, so we decided to apply this question to a specific biological question, which is the evolution of human brain. So brain is a big part of what makes humans human, and specifically the cognition is the most important aspect of our brain development. So there is recently there was a data on embryonic human and macaque brain at multiple time points. Specifically, they profiled not only the gene expression, but what's this specific epigenomic mark, which marks active enhancers in the genome. It's dynamic, it changes from context to context, cell type to cell type, but that's what marks the current state of the enhancers. Okay, so we trained our model, and this is simply showing whether given a random sequence, how well we can classify that as enhancer or not enhancer, okay? And this is simply showing that when you train it on human, how well it does on macaque, vice versa, all these kinds of cross comparison, cross validation. So at least the first task, which is given a sequence, how well we can tell, is it enhancer or not, it can do reasonably well. But the second question is even more important, which is how well can it distinguish the effect of a single nucleotide change? So for that, the experimental validation is hard. So we relied on an existing RAQTL data, which is a large-scale reporter assay-based determination of single nucleotide differences in the enhancer activity using reporter assays, right? So the question here was, if there are two alleles, right, A and B, if our model says A is more likely to be enhancer than B, how often does that agree with the experimental data? So the difference between our sigma, the final, the score, the larger that score, the more confident we can be that we are pointing to the right allele. So that's why we did this kind of analysis. The greater the delta between the two scores, what is the accuracy? Meaning, what fraction of times we pick the right allele according to the RAQTL experiment. So you can see very quickly this kind of delta, it reaches almost perfect accuracy. So we were confident that this model not only works in terms of identifying enhancers, but single nucleotide change is sensitive enough to pick out the differences at single nucleotide level. Okay, so with this model, we went ahead with our fundamental question we were after. And this is what we did. So we got human, we got macaque. These are the enhancers, the K27AC. And we are interested in knowing which enhancers were gained during human lineage evolution and which enhancers were lost. And if you simply see the difference between the two species, you cannot tell whether it's gained or lost, right? So we need to go back and figure out an outgroup to really ascertain gain or loss. So that's where this outgroup determination comes from. So we estimated the sequence of the outgroup sequence using some probabilistic arguments. So this is established in the field, but we don't have the experimental data because it doesn't exist, this species, but we have the estimated sequence for this outgroup species. And we use multiple criteria to say whether an enhancer is gained or not. Here are the criteria. First, you see a K27AC peak in human and not in mouse, not in macaque. Second, our model says yes, our model says no. And our model also says no for the ancestral species. If these three conditions are met, then we call it a gain, and just the converse of that would be loss. Is that good? So we got about 4,000 gained enhancers and 3,000 lost enhancers. And next step was identifying the specific mutations that might be responsible for this gain or loss of enhancers. And how do we do that? For every enhancer, we have some number of mutations, differences between macaque and human. We start with the macaque sequence. We introduce one mutation at a time and see at what point the enhancer model says, now it has become enhancer. Okay, so, and as soon as we get that, then if a single nucleotide change goes from no to yes, then we call that nucleotide mutation essential mutation. Okay, so it turns out that about 40% of these 4,000 gained enhancers, according to the model, could be attributed to a single nucleotide change. Okay, now what else did we learn? So this was just the beginning, first 20% of the paper, rest 80% is just doing all kinds of analysis to hopefully convince a neuroscientist or biologist that this has any value, okay? So I'm just going to summarize all that in one slide, 2 years worth of work. So what are the salient properties of these gain enhancers with essential mutations? So first thing we noticed that these gain enhancers are enriched for cognitive trait GWAS signals. The genes near these gain enhancers, they exhibit an increased expression in human embryonic neocortex, and the lost enhancers show a decrease in expression of the nearby genes. What else? If you look at the genes that are involved in critical neural developmental processes, then we went to a single cell brain, human brain single cell data to ask which type of cell these genes are expressed in, because that's an important question to really ascribe function to them. So when we do that, we notice that there are 2 types of cells these genes are preferentially expressed in, and those are neuronal progenitor cells and interneurons, which make the connection between different types of neurons. Okay, so that was to us very significant. And lastly, we did a very complex motif and various analysis to figure out this thing called co-regulatory network. So for example, you may have a POU3F2 binding site in an enhancer that is gained and has a mutation, and the target of that enhancer is POUF3 itself, right? So the autoregulatory loops. So all these arrows here are those types of things where you see an enhancer that is bound by something, but is regulating something else. And that in turn is regulating all these guys here. So this is the standard definition of what we call co-regulatory network that maintains a cell type identity. So ultimately, we identified this co-regulatory network with POU3F2 sitting on top of the hierarchy, and all these transcription factors are very well established in terms of brain development. So that was the bottom line of this project. How much time I have? 2 minutes. Perfect, perfect. So that was just a setup for, because I'm at NCI, but this was important for me to set up the tool to be comfortable that this tool actually works. But now moving on to cancer, it's a whole different level of problem here. So ultimately, we want to figure out 2 types of things. First, in the cancer field, first of all, most of the focus is on mutations. And out of that, almost all the focus is on mutations in protein coding part of the genome. What about everything else? That's where I'm coming from. So nobody is doing this kind of work. So, and for do this kind of work, you need specific type of data. You need tumor, and you need in the same individual, the adjacent healthy tissue, to be able to be sure that there is a mutation. Okay, so we got this kind of data in 10 esophageal cancer patients where we have tumor and we have adjacent healthy, and we just do exact same exercise. We develop a model, we look at the differences in enhancer activity, we look at the mutations that fall in there, and then we do biological analysis to see what they might mean. So this is part of the summary. So enhancer gain and loss are associated with increased decrease of gene expression. That's the bottom line. Gained enhancers are associated with inflammation, innate immunity, or regulation of adaptive immunity. That was very interesting to us. And we are seeing this in multiple cancers at this point, these 2 signals. And the lost enhancers tend to target genes involved in differentiation. So our general big picture theory right now is you lose the enhancers that are maintaining the cell type, the cell is becoming more dedifferentiated, which is the basis for beginning of cancer. And gained enhancers are keeping the immune system at bay at the same time. So these 2 things go hand in hand. And then we use the K27AC data directly to identify mutations. And first of all, we show that the mutations are enriched in the lost enhancers, which again makes sense. It's easy to break things than to make things. So typically, mutations will break enhancers. And when we look at the gained enhancers with mutations that are even more enriched for the specific processes I just talked about, which tells us that there is actually a selection going on. Otherwise, you would not see any enrichment for the enhancers that have mutations in them, right? We also found that the gained enhancer genes are enriched for higher hazard ratio. So they are They are pro-tumor in the TCGA data, completely independent data set. And they're associated with, a repressive adaptive immune system based on this TIDE score. And lastly, last slide, the lost enhancers are again with mutations are even more enriched for this differentiation type of genes. And they are enriched for tumor protective genes, anti-tumor genes, so which is hazard ratio less than 1. And this is a very high enrichment here. So again, everything falls into place. And so we are continuing to do this and trying to collaborate with people working on esophageal cancer to go further. And we have a second project where we are interested in looking at, there is a big difference, population differences in various cancer incidence rate and severity. So it must be encoded in the polymorphism somehow. So that's the first, thought. So how do we figure that out? So we are going through all the polymorphisms that have, so we are focusing on prostate cancer because, African Americans and whites have very big discrepancy in the incidence rate and severity. So we are looking at polymorphism genome-wide that have very high FST, which means they have very different allele frequencies between whites and African Americans. And then we go through our series of filters. Are they encoding for enhancers? Do this polymorphism change the enhancer status from yes to no, no to yes, this sort of stuff? What kind of genes they are near? So again, this is showing a lot of interesting results so far, consistent with what we are seeing at the broad level with the esophageal cancer. So they're very much in the early stages, but I'm quite excited about the future of these projects. So last but not the least, Sean Lee, staff scientist in the group, did all the work that I've presented today, and special thanks to our admin staff.
Any questions for Sridhar?
Of course.
That's why Soren brought me here. Anyway, extremely impressive.
Thank you.
So can you detect synergy between enhancers?
So that requires a whole other level of data, like more data and more phenotypic, like clinical properties of this. But at the level of, okay, if my phenotype is gene expression, let's say like EQTL, for example, then definitely we can do things like this, what you're suggesting. Look at pairs of things. At least for a given gene, we can look at all the enhancers in, let's say, 1 megabase proximity, and look at the combinations of these alleles and see if they have synergistic effect in terms of this gene's expression. But if you go to the real phenotype, which is cancer incidence or clinical thing, then I think data might become a problem. In principle, yes, but in practice, I don't know.
And the other similar question, is there any correlation between mutations and enhancers and neoantigens?
That's a very good point. Yeah, so our focus is entirely on how these guys might change gene expression, not how they might be expressed and present as a neoantigen, which is what you're alluding to, I guess. So we haven't even asked that question yet because we are focusing on regulatory region only that typically are not expected to make peptides. So I don't know. Yeah. Thank you.
Thanks again, Sridhar. So I'd like to introduce our next speaker, Jonathan Udell. As long as I'm giving Celera anecdotes, I wanted to mention that Jonathan is part of a very curious and I think not well-known part of Celera's history, and Sridhar actually set it up perfectly. So after the genome was sequenced, I think there was some casting about for what Celera does next. And I have no idea how the decision was made, but the decision was that we would be working on cancer immunotherapy. And this was in some ways, a very far-sighted decision because it was really a pie in the sky project then. And today, cancer immunotherapy is the hottest thing in cancer. but it was too far-sighted because it was about a decade after Celera went away that it actually, the major problems, checkpoint inhibitors, were solved, and it actually became a useful technology. But it led to some cool science, and it let those of us who are there say that we were doing cancer immunotherapy before it was cool. So we got to work with Jonathan and Steve Hoffman and others, and I hope you, that is, of course, one very small part of what he does. He's really a giant of the field of immunology, and of course, it was recognized we couldn't do this without an actual immunologist, and we're really happy to work with him, even though we never cured any cancer. So thank you.
Well, you could see where Celera was heading if they had to consult with me to make money. So that was certainly a harbinger. Okay, so now for something completely different. I know many of you are, you know biology only as much as you get sick or are worried about COVID and relatives, et cetera. But I'm gonna tell you some immunology. There'll be like no data. If there's a little bit, I'll describe simply what it is. And we're going to talk about the immunopeptidome. So very good introduction. The immunopeptidome is something you need to know if you want to specifically target cancers with T cells. I have a subtitle. There we go. Making drips great again. And so you're wondering what the fuck is a drip? I will tell you what a drip is. It's not a person, although it's both actually, right? Okay, so T cell immunity. There are two kinds of T cells. You have the helper T cells, which is what get boosted when you get the COVID vaccine. You need the helper T cells to make antibodies. Without helper T cells, you don't make a good antibody response. There's another kind of T cells called the CD8 T cells. CD8 is just a cell surface protein. That's a differentiation marker that's very convenient. And these guys originally were called cytotoxic T cells. This is what won a Nobel Prize for Zinckernagel and Doherty in 1996, because these cells play a really, really important role in viral infections. They don't prevent viral infections, but they make sure you don't die from them. COVID would have been much, much worse if SARS-CoV-2 wasn't related to other coronaviruses that we get routinely. There were 4 circulating coronaviruses. We have a 5th, and now some of the other ones are probably going away. The T cell determinants that are recognized by the helpers and the killers are conserved, many of them. So we were not a completely naive population. Think about that. 1 million dead Americans with vaccination, right? Not all vaccinated, of course. The number would have been much, much higher. Probably would have been 1918 flu-like had there been no cross-reactive immunity. Okay, but there was some. So these are really important. And I've spent a good chunk of my career studying CD8 T cells. What do they recognize? They have a clonally restricted receptor called the T cell receptor, or TCR. You have, 10 million plus different clones floating around in your blood right now. Most of them will have completely unrequited love in their whole careers as T cells. They'll never find a target cell, but the lucky few ones find a target cell and then they proliferate like crazy. We have a really cool paper in eLife where we actually are counting. You guys are quantitative. We count the proteins in the cell and the ribosomes, and we looked at T cells because they pose a really intriguing biological question. They can divide every 6 hours when they're cranking. How the hell can they do that? Right? That's a lot of protein synthesis. And it's a cool paper because we have a paradox. We try to do the numbers as carefully as we can, and this is not the numbers you guys do. This is what you learned in first grade. Immunologists are capable of that, most of us, which is just counting. We count proteins, we count ribosomes, we can measure the rates of translation. They shouldn't be able to divide as fast as they can, and yet they do. So we didn't answer that paradox. Okay, but that's something else. So I'm going to talk about, wait, I got to go back. I'm sorry. I'm going to fly back in here. The origami's coming back. What the TCR recognizes something called an MHC class 1 molecule. There's a really cool molecule. This molecule evolved to take a peptide that's made inside the cell that's, say, 8 amino acids to 12 amino acids, and it carries it out from inside the cell to the cell surface. It's a phenomenally sensitive system. A T cell can kill a cell expressing 1 or 2 copies of the peptide, right? Out of 100,000. TCR sorts through all these and then makes literally a life or death decision whether to kill the target cell. So These are really important in viruses, and it turns out Celera was ahead of the curve. Absolutely. They are also very important in cancer. The way the checkpoint inhibitors work, at least 50% and probably more, is by unleashing the T cells. Cancers are not stupid, right? By the time someone shows up in a doctor's office with a cancer that's clinically presenting, the cancer has evolved to evade the immune system. There was a very long debate in immunology whether cancer immunosurveillance even exists. It does. One of my college roommates is living proof of that. He would be dead. He presented with like 4 pounds of melanoma. He was one of the poster children for checkpoint therapy. Within 6 months, there was no detectable cancer. So when it works, it is unbelievable. Dead people are walking. Field of antigen processing, fantastic field I helped get started in the 1980s. Lots of great cell biology. There's a pathway, there are textbooks. All the rules are breakable because it's biology, it's not physics or math, okay? So everything you read is probably a little bit true-ish, right? It's one thing you guys need to know if you haven't already learned about biology. Things that look like facts, at very best, they're sometimes facts. And there are always exceptions, always. And this is just a classic example. Everything I'm showing here is way oversimplified. Basically, you have a protein. It's a great cartoon. It's a chicken because we all work on a chicken protein and chicken peptide, which is stupid, but we do. It's a very useful system. The protein gets degraded by something called the proteasome, which is this, Cuisinart for cells that can basically degrade every protein in the cell into little bits. Some of the bits make it into something called the endoplasmic reticulum, which is the inside part of the cell, where you assemble things that are destined for the outside part of the cell. And what happens here is the MHC class I molecule is assembling with the peptide that's going to carry out to the cell surface, where the T cell is going to decide whether to beat the cell with the pipe or not. Okay, so this is the class I antigen processing system. The repertoire of all the peptides that are presented, and there are millions, is called the immunopeptidome. Where does that word come from? It comes from this paper, because we invented this. In helping destroy Celera as a financial enterprise, we invented the word immunopeptidome. And this is a really great paper, and thank God that somehow you guys found me. I don't think I've ever had as much fun in my career, because to be honest, this was my take on it. Steve Hoffman was clueless, right? Completely clueless. You had all these incredible brains, and you needed a guiding rudder. And I have, as Soren knows, anyone who knows me, I have more ideas than I could ever follow, like most scientists. And this was working with the dream team. It was just absolutely the dream team. You guys were so smart. I would have an idea. 2 weeks later, there it would be. And we were the first ones to apply informatics to this question of where do the peptides come from and what is the evolution? And I think my favorite experiment in the paper, the question was, okay, did the proteome, did the genome evolve to favor or disfavor peptides from being presented? And this was pre-Celera, but there were already decent algorithms. They've gotten much better for predicting. It's one of the early examples of where machine learning worked in biology for predicting peptides that would bind to class I. These rules were pretty good then. They're a little bit better now, but they were good. So we did this study where we could predict peptides and whether there was evolution for binding or not binding class I molecules. And my favorite organism was the vent organism. Right? We took one of these organisms that evolved at the bottom of the ocean at huge pressures and gigantic temperatures, and it turns out they were no more or less liable to have a binding peptide than we are. Right? So there was no selection for or against peptides. That was what I love about informatics is when you answer a question like that, it is definitive. Everything else I do, it's not definitive. Because even if your evidence looks great, as a biologist, experimentalist, you're always worried. Did we do this? Did we do that? Did we pick the wrong salt concentration? Did we not? Why didn't we do this control? You do something with informatics, okay, maybe it's completely wrong, but you don't have to worry about the method, right? Just maybe you have to worry about your assumptions, but not the method. Okay, so I got to know Soren, and we had, for a while, we had a pretty close relationship because he was a cool guy, incredibly brilliant, and I had lots of ideas about what one could do with informatics, and he invited me to Brown. Just downstairs. And at that time, I was already giving this talk I give for young scientists on how to succeed in science. And I gave it to a group of Soren's people, and I found out it was actually useful Right, so that's for an informaticist, that was really quite an amazing thing. So thank you for that last night. I didn't realize that. Yeah, and it's been online ever since. And I think a lot of kids have seen this video. So thank you, Soren. And I hope Brown keeps it posted, for forever, basically. I'm still giving the same talk, only it's not as good because I'm really slowing down.
Derek was the one who put it.
I still manage that account.
Thank you, Derek. All right, we have to keep Derek alive. Yeah. Yeah. Make Derek great again. Yeah. Okay, and that talk led to a couple of essays that I'm happy to send to you. I think these have been downloaded hundreds of thousands of times about how to succeed in science. And then I eventually took a sabbatical where I went there not to write the book, and eventually I wrote it, right? So there's a book. I wrote this as a government employee. You're not allowed to set a book at 0 on Amazon, right? So I'm actually breaking the law because it's for 99 cents. But if you don't want to pay 99 cents, email me and I will send you a link to a Google folder where you can download the book in an e-format. Okay, and it's a useful book. It's very practical. Like the books I read when I was a student, they just pissed me off. There's a famous one by a guy named Peter Medawar. The title of it could have been, "I'm smart and you're stupid," right? So my book is not like that. It's just, what does it take to get through graduate school? What does it take to do an experiment? It's very pragmatic. Okay, so what are DRIPs? The DRIP hypothesis from 1996 was proposed by me basically to explain a curious observation. You take cells, you infect them with a virus. You have a trick for stopping protein synthesis various times, just a drug. Okay, you do that, you find out 30 minutes after the virus has attached to the cell is enough time to make enough protein that the T cells can kill the virus infected cell. Whoa, that's really fast. Makes sense. Viruses are very fast. SARS, not so fast. Some viruses can replicate in 4 hours. SARS-CoV-2 in vitro anyway takes a day or two, but still fast, right? So that is really fast. Okay, so this part didn't make sense. The peptide, even at that time we knew what the peptide was, it comes from a protein whose turnover you really can't measure because the cells die. So the average cellular protein has a half-life of a day or so, right, on average, across the whole proteome. For viruses, the proteins are at least that stable on average. They're really stable proteins. Okay? Another problem. You have, in 30 minutes, you have maybe a few thousand copies of viral proteins. How many copies of host proteins in a single cell? 2 to 3 billion, something like that, for an average size cell. Billion. So 100,000 class 1 molecules, you guys can do the math in your heads. That ain't going to work. If we're going to wait for degradation, there's no way. Right? The kinetics they're not going to allow it. So hypothesis is that to enable immunosurveillance, a virus's peptides derive from something that we call the DRIP, which is a rapidly degraded defective ribosomal product. So I didn't want to dignify the substrate, as a protein because we didn't know what they are. We still don't know what they are. And I wanted to leave open the possibility that it was just like a shorter peptide. Right? So I called it something that wasn't protein in the name, a defective ribosomal product because it is a product of the ribosome, right? All of you know the ribosome is the thing in the cells. To me, evolution's greatest achievement, making this machine that produces all of the proteins on Earth, something called a ribosome, right? Every cell has to have them. So here's the idea, right? So here's another cartoon from a friend of mine, Eric Wrights. We're making the chickens, the model protein, they're coming off. Some of them are mistakes. Maybe they started in the wrong place. Maybe they finished in the wrong place. Maybe there was a frame shift. These are headless chickens. These protein synthesis errors in protein folding, which is not perfect, very imperfect, right? Sometimes. All these guys are going to be triaged to the proteasome. We're going to make peptides, and these are going to be presented to the immune system. Okay, so that's the idea. That's the simple idea. And for viruses, this turns out to be really, really true. So this is the only data I'll show you. So here, this is mass spectrometry, not by my lab, by a very good lab in Australia, a guy named Tony Purcell. And this is influenza virus. infected cells. We're looking at influenza virus proteins. The names don't matter. The red is the protein, okay? The grayish thing are the peptides from the protein. Look at this. Sometimes they can detect the peptides before the proteins. As I said, all of these proteins are stable. This protein and, is that any there? And this protein are infinitely stable because they're exported, right? So they go outside of the cell, so they don't even count. So in that case, the peptides had to come from drips. But even from the other ones that are not exported, that are staying in the cytosol, in the nucleus, the peptides are being made basically co-translationally. So this is confirmation of the model. Okay, and this has also been done for vaccinia, but for no other viruses yet. The mass spectrometry is not easy. And you have to be a lab dedicated to it, and it's costly.
Y-axis on the previous slide?
Sorry, I should have explained. This is just a relative amount. Yeah, and it's not super quantitative either, which is one of the problems. If you want to quantitate the peptides or the proteins, not so bad. Peptides, you have to make a standard curve with a known peptide.
And do these peptides come from throughout the protein or only from the big?
No, they come from, that was something we actually looked in this original immunopeptidomics paper. They come from throughout. There's some biases that we still haven't figured out, but more or less it's from the whole protein. Okay, so what are they? When you make a full-length protein, what are the peptides, or when you have a full-length gene? So everything you could think of, every possible error in literally translating information from the original nucleic acid to the protein. It's proven difficult to nail down biochemically what they are, but after 27 years, here are 3 examples of defined influenza virus drips in ascending order of craziness. Most of these just found in the last 5 years, some in the last couple of years. One is in one of the gene products, it's a +1 reading frame, starts on a methionine, peptide starts there, 14 residues, right? So something that small, anything less than 50 residues is probably not going to fold. So unless it finds a partner, it's going to be degraded. Something that's 14, it's not impossible that it has another biological activity. There are some peptides that small that bind something that have activity, but this is probably just a mistake by flu. Okay, influenza virus. There's another one. It's a model peptide, but still tells you what can happen. This is a crazy one. It's in a +1 reading frame. It's after a stop codon, and it's initiated not by AUG, but CUG. So what antigen processing tells you is that all the things you think you know about what it takes to make a protein, you have to expand them. Do you mostly make proteins on AUGs? You do. But actually, that rule is really coming into question now. There's more, if you're just counting gene products, not abundance of proteins, more than half of the gene products, way more, are initiating on non-AUGs than AUG, right? So that rule is really not a rule. And then here's the craziest one. We published one example. We now have 5 other examples where we are making peptides from the negative strand of a negative strand virus. Okay, so what a negative strand means is it's non-coding. So whenever I use the word non-coding or UTR, I have to make air quotes, because a lot of the things we think are not translated, they are translated. And I'm not telling you it's abundant translation, but I'm telling you the immune system thinks it's interesting. And the immune system can use this for immunosurveillance. Okay, what is the overall contribution of drips to the cancer immunopeptidome? So we had a really good collaboration with a very good friend of mine at NIH called Lou Stout. Lou is the world's expert on human B-cell lymphomas. And we took three of the human lines that Lou has developed. We did ribosome profiling. This is a method worked out by Jonathan Weissman, incredibly difficult, incredibly powerful, right? This is a method after $10,000 and with a really good postdoc and doing it 6 times, you can figure out everything a cell or a group of cells is translating. It's an amazing experiment, amazing method. And then we had a very good collaboration with an excellent informatics and peptidomics group in University of Montreal. And this was a really nice paper in Cell Reports. My lab did the ribosome profiling. The other guys did everything else, including the bioinformatics, which in this paper I thought was quite amazing. So the RiboSeq data, this revealed 181,811 non-annotated proteins, in addition to the 26,000 plus canonical proteins. So Craig was saying the other day that the 400,000 number was wrong, but maybe not as wrong as you thought. And not for reasons you ever could have anticipated. Because these are things that, again, by definition, they are non-canonical, right? So, but there are a lot of them, and this is not the only paper. The original paper from Weissman, they were already showing there were tens of thousands of non-annotated genes.
These are short peptides or actual
You could, yeah, there are all sorts of things, right? But things you think are UTRs, they're not necessarily. A lot of them are translated, right? And then this is what we're detecting as well. And even though you're getting what seems like a lot of reads, right? 500 million reads, that's 100 cells worth of ribosomes. It's nothing. So God knows in a cancer what exactly is being translated, right? It's a lot of things. So mass spec, not as sensitive as RNA, DNA-based methods. This is a good group, 14,500 proteins they're detecting. 17% are non-canonical out of our 181,000. So 28% are splicing variants, 72% are cryptic, UTRs, frame shifts. The cryptic proteins are predicted by bioinformatic algorithms to be more disordered, unstable. They're drippier, according to us. And this we could do per translation event, because that's what we're counting by ribosome profiling. They are more likely to create a detectable peptide, right? So that all kind of makes sense. Okay, so thank you, Bill Barr. We're breaking the law here, right? How does the immunopeptidome circumvent the law of mass action to prevent the most abundant proteins from dominating the immunopeptidome? 250 proteins account for 75% of the proteome, 250. These are the housekeeping proteins, molecular chaperones, ribosomes, translation factors. The most dominant proteins are a huge fraction of the proteome. If it was just a come-as-you-go system, the immunopeptidome would be dominated by them, and it's not. And you wouldn't be able to see viruses. So we're breaking the law of mass action in terms of not every protein that's made, they're all going to be degraded, winds up with an equal chance of being a peptide that's presented. How does that happen? We had an idea a long time ago, 20 plus years ago, a real unicorn for my lab. The immunoribosome hypothesis is that a subset of ribosomes generates substrates at high efficiency for immunosurveillance. So the idea is you have 3 million ribosomes, maybe 10,000 of them are actually really good at making peptides for some cell biological reason. That was the idea, right? So the immunoribosome makes a drip, which goes right to the proteasome, which goes right to the transporter, which goes right to class 1. That would be a model of something that's what we call channeling in cells, right? Many enzymes are known to be channeled. You don't randomly have a substrate going and a product going from one enzyme to the next in a metabolic chain. You organize the enzyme so it's more efficient. That would be the idea. Okay, so it was proposed in 2002 and not widely for you guys, but widely for my tiny little field. My 15 colleagues who knew about it thought it was a stupid idea, right? That was widely ridiculed for this field, right? Okay. But what I love about science, which is my arrogant name of the book I wrote, is truth wins, right? I started as a history major as an undergraduate, and it was like whoever was the best bullshitter won the argument. And in science, I'm not saying the truth, I think, as a biologist, is something we approach asymptotically. We never get there, but you have evidence, right? So if you're right in the end, you could say to the world, fuck you. Right? That's the great thing about being a bench scientist. If you're right, you can show it eventually, or somebody can, right? Hopefully your career won't be over by that time, but that's one of the advantages of being in the intramural program in NIH. We're not like Elliot Ness's untouchables, but close, right? So it's hard to fire us, right? So it's good to be there. You can have big ideas and just ignore people when they criticize you. You don't have to convince the study section. It's not a completely stupid idea.
That's why ESTs work there.
Yes. Okay, and eventually I got lucky. I got a great person. My lab postdoc named JJ Wei, who's now at Merck. and JJ was the unicorn. And what JJ did was knock down every ribosomal protein and say, does this affect antigen presentation? And it did. Okay, we could upregulate antigen presentation, we can downregulate antigen presentation. It had nothing to do with how much protein was being made. Okay, we did this really nice. We had no idea how it worked. We had no idea how important it was. But is this relevant? It turns out we got really, really lucky at the end of this, which is why the paper's in Molecular Cell, because these knockdowns of ribosomal proteins are what cancers do. Very few cancers that don't upregulate their ribosomes or downregulate or even get rid of some of ribosomal proteins. So the ribosome is mostly an RNA machine, right? And the RNA itself can be a shitty ribosome, but the proteins make it a lot better. There are 80 of them. Okay, we found out you can knock down 20 of them and have an influence on peptides. And it also turns out that a friend of mine at NIH named Nick Restifo had just published a paper in Nature where they did the first CRISPR screen for how tumor cells can evade cytotoxic T cells. And oh my God, one of them, where's the smoking gun? It's pointed the wrong way, so the slide got screwed up. But RPL23 is a ribosomal protein, this guy up there, right? So there it was. Could this be relevant? JJ started going to their lab meetings and doing experiments and hanging out with them, and yes, true. So what JJ discovered was that tumor cells can downregulate T cell recognition by getting rid of ribosomal subunits. So the immunoribosome hypothesis, while it may not be generally true, it's certainly true for cancer immunosurveillance. Okay. So that's the talk until the, what was it you asked for, Soren? What was it called? Open problems, right? So everything I just told you is an open problem. But here are some of the more generally interesting ones. How is the law of mass action circumvented by DRIPS, physical channeling? Okay. How do immunoribosomes work? We really don't know. Are there natural immunoribosomes in non-cancer cells? We don't know. What is the efficiency of protein synthesis in living animals? It's a really important question. On the way to DRIPS, we discovered that the efficiency of protein synthesis in cultured cells, including normal cells right out of an animal, is nowhere near 100%. It's like 70 to 80%. And that's a disturbing number because protein synthesis is the single most important energy sink in a living organism. And something like 5 to 10% of the bagel you ate today went to maintaining your proteome. And any tiny selective advantage in efficiency would play out in evolution, right? Because starvation is a thing if you're an animal. Look in your backyard, they're starving, right? All day, they're just trying to eat. Okay, so what are the errors that account for this? Something that comes out of this translation work, these error numbers, which is something that my lab helped get started, is basically showing that proteins are statistical. And a 500 amino acid protein, there's a very good chance there's a change, not a genetic change, an error, right? So proteins, you think of them as the protein, they aren't. They are they're a swarm of different protein species. They have different amino acid sequences, 1 or 2 changes. And on top of that, of course, conformationally, they're never one thing, there's a buzz. How many gene products are there really, and what do they do? What are the biological functions of degradation products of the proteasome other than immunosurveillance? There must be some, because they're really abundant. Why use alternative start codons for non-canonical proteins? Why not just always use methionine? Are there reasons? How often do they result in non-methionine initiation? Classically, even if you have a CUG, you use a methionine. A colleague of mine died, sadly, tragically, a couple of years ago, Neelabh Shastri, showed that sometimes you actually decode CUG, which is the most common non-cognate codon, as a leucine and not a methionine. Why? Why do you always use methionine? Why is methionine the initiating residue anyway? Something else, why are there 274 plus different human tRNAs? What the hell is going on? There's only 20 amino acids. There's only 64 codons. We only use 47 anticodons. And yet we have almost 300 tRNAs. What is going on? So many questions there. And I will end there.
Any questions from the chat?
So it's very impressive what you're showing and what we've known for a long time from trying to model cells and create unique cells. Is almost a quarter of the genome in a minimal cell is devoted to cleaning up all the garbage in the
Yeah,
cell on
that's
a
right.
continuing basis.
That's right.
So all these systems are sloppy. Why are they sloppy? Because the binding affinities of all these things are part of a Poisson distribution and a spectrum.
Yes.
And as I said yesterday, so much of discovery is overcoming the bullshit ideas from the past. And why we ever assumed that these were high fidelity systems, it's not there. So, but the question I have is the immunosurveillance a byproduct of the sloppiness in the ribosomes, or is, as you suggest, there is a subset of ribosomes specifically for that?
Both is what I would say. If you look at something like yeast, which don't have immunosurveillance, and there's only one paper on it, and then we did some experiments we didn't publish, and you look at the error rate in protein synthesis in terms of what's rapidly degraded. By rapid, I mean something like a half-life of 5 minutes or so. It's the same or worse than HeLa cells, right? So this looks like it's possible. You know, evolution is not about being perfect. It's just about when the tiger is chasing Craig and me, I have to be faster than him. And right, so it just has to be good enough.
Only if we're running in the same direction.
In the same, yeah. Or we get, yeah, as long as the tiger can't split, yes. Yeah, so I mean, it is entirely possible in the end that that's just the best evolution can do. But I would just say, in general, it's really dangerous when you have a good idea and then you have evidence supporting it. When you're a PI for a long time, these are the things that make you really scary, particularly when you're not doing the experiments yourself. And it's just very convenient to fall into what you think you know is true. And so it's just one of these things we don't know, and it's something that I think is not so easy to answer.
So one of the slides I made when I was asked to give the Schrodinger "What is Life" lecture, second one to him doing that, there's a number of autoimmune diseases, as you know, that are defects in the systems for taking out all the cellular garbage. And the slide I was at just New York City during a garbage strike, because that's what happens inside the cells.
Yeah. Yeah.
What turns on the drift upon viral infection?
Yeah, good question. Right. So innate signaling really increases the peptide supply, right? And the drip we made for immunosurveillance, but there's something, this other thing which people are calling drips was not the original intention that I had, but these other defector proteins, many of which are somehow not contributing to the proteome. Yeah. So something with innate signaling actually is turning on, something Neil discovered. Yeah. But we should sit down and talk sometime. And we're just like neighbors and.
So our next speaker is going to be Daniel Fasulo. He was a key member of the assembly team at Celera Genomics and has gone on to do many interesting things since then. I haven't seen Dan in more than 20 years, so I'd be as excited as everyone here to learn what those exciting things were. But Dan will be telling us about the microbiome and diabetes. So, okay.
Thank you. That's good. Where's this? Okay, so before we get started, I would like to say happy birthday to Soren. As I sat through this conference, I came to a realization. I hadn't really thought about this before, but the very first conference I went to as a shiny young grad student, second year, was Recom 97 in Santa Fe. And at the time, it was all new for me. It was, a completely new experience. So it didn't really stand out to me how special that conference was for everybody else. I just assumed that for them, it was all normal. But as people still continue to talk about that meeting, I realized what an effect you had on me even before my days at Celera. So thank you for that. So yes, briefly about myself. I was one of the very few Solera informatics people that decided to stay in the corporate world after Solera. I worked at Siemens Corporate Research in the healthcare research division for a few years. And then I got excited about next-gen sequencing. So I went to 454 and managed some informatics groups there. After 454, I was independent for 10 years doing bioinformatics software development and consulting. I had a few ideas of my own I wanted to pursue. And at the same time, I was helping companies, including a company called Shoreline Biome, which produced a kit for 16S sequencing on next generation sequencers. And just this past year, actually, they decided to change their mission to become much more data analysis focused. And they merged me into their company and formed Intus Biosciences. So I am now head of bioinformatics and IT at Intus Biosciences, which is a small startup in Connecticut. And just very briefly, this is mostly going to be an algorithms talk or data analysis talk, but just so everybody's on the same page, I want to talk about what the data is. So every bacterium has a 16S ribosomal RNA gene. People will often break it up into regions V1 through V9. Even back in the Sanger days, people would capture this and sequence it because it's highly conserved, so you can get primers to amplify it selectively, but there is variability in it. And so you can determine at least the family, sometimes the genus or species of a bacterium by looking at this. Now, when next-gen sequencing came along, there was a dilemma because you could get a lot more reads. That would make you a lot more sensitive to what was in the sample, but these reads were short. And because they were short, you couldn't capture much variability. Therefore, you couldn't really tell what you were looking at. So what some people did was propose shotgun metagenomics, where you just sequence everything in a sample. You try to restrict it to the bacteria the best you can, but then you sequence it all and try to make sense out of it. And the problem with that is it's very complex to analyze, and also you lose some sensitivity now because you're sequencing the entire genome of everything that's in there. So the Shoreline Biome guys call this the Goldilocks approach. Now that next-gen sequencing is returning to longer amplicons, we can sequence not only 16S, but we can design a primer in a name-bearing gene called 23S. Not all bacteria, but the overwhelming majority of bacteria have a 23S gene proximal to 16S, and you can sequence through the whole thing with an amplicon of about 2,500 base pairs. And what this gets you is not just the 16S gene, which we already have some good databases for, but the ITS region, which is hypervariable. So you now have a chance to identify and track very specific types of bacteria through this region of this amplicon. And they did a little preliminary work on the right side of the slide here, where they show the longer you sequence, if you've got 150-ish different types of E. coli, the more likely you'll be able to find something in that amplicon that separates them. So this basically says with the Intus Bio amplicon, you can accurately separate all these different things. Today, I'll be talking about a data set that has already been published by some collaborators, and they were looking at diabetes and the gut microbiome. So these are fecal samples. And they stratified, they had 47 samples, not a very large study. They stratified it by recruiting patients that were healthy controls or that had been diagnosed with prediabetes or diabetes. And when they enrolled them, they did their own A1C test on these patients to confirm the stratification. And what they ended up with was 4 levels. They had 21 healthy controls. They had 8 pre-diabetics and 10 pre-diabetics. And then they had this sort of block in here, and I'm not quite sure what to do with these. These people were previously diagnosed as pre-diabetic, but it may have reversed, or it may have been just right near the threshold because they tested as non-pre-diabetic at enrollment time. When I talk about our analysis, by the way, I took the 10 diabetics, and instead of looking at mismatched group sizes, I decided to population and gender match and go down to a 10 versus 10 comparison. So what did they do and what did they find? Let's start there. They extracted DNA from stool using a QIAGEN kit. They used our kit to prepare these samples for sequencing. So this creates that amplicon, and it also attaches multiplex identifiers to all the samples so that you can separate them after sequencing. They used a PacBio Sequel too. Of course, we are experimenting a lot with Oxford as well, now that it's finally getting to the point where it may be practical. And then they took it into our software package where they were demultiplexing it, doing some basic quality control and error correction. And then finally, they took all these reads and they mapped them. And we have a database generated from genomic sequencing. There are 16S databases out there, but there aren't a lot of databases that have our entire long amplicon sequence. So we have basically done our best to try to generate our own that people can map to. And then the very end of this process, you can imagine they have a matrix, they have their samples, they have the taxonomic classifications of the reads, hopefully, some of them, most of them. And each cell of that matrix is just a count of how many reads had that label. And so you can take this now into a framework like R and do a statistical or machine learning type analysis on it. And indeed, what they did is they used some packages that convert this so it really looks like gene expression data and put it through DESeq2. And what did they find? They found several species of Bifidobacterium that were populous in healthy individuals. Rhomboutsia is downregulated in diabetics, so also more populous and healthy. Some different types of pathogens, Escherichia coli, Klebsiella, more common in diabetic individuals. And these are in green because you'll see in our analysis that we recapitulate these results with our analysis, even though it's quite different. They also found a couple other species. What was kind of interesting is we did not recapitulate these, and they note themselves that their finding is inconsistent with the literature, which means that when they looked at the literature, people either predicted the opposite or they saw no effect when they did similar types of studies, et cetera. So what is the issue with this? Why am I talking about this as opposed to just saying, okay, they ran some tools? Why do I say we need new algorithms? Well, if you think about what's going on here, there is an enormous chain of algorithms that gets run on all of these reads. And all of them, particularly the identification, can make mistakes. So by the time you're getting this into the analysis package that's going to tell you the question you really care about, what's different between diabetes and healthy individuals, your data has been processed. It may have errors, it may have omissions, it may have biases from all of the processing that you've done up front. And what we want to do is we want to really make answering the question about diabetes the core of the analysis and annotating what that core finds secondary instead of annotating first. So we have a workflow that, again, it's identical up to the point where you separated everything by sample, the reads by sample. But then we send things into a series of software packages. I don't have time to go into huge detail. Luckily, they're all relatively simple, so I can sort of talk through them, and I'll have another slide with some pictures after this one. But the idea is I have all my demultiplexed reads by sample. I have my sample metadata, particularly diabetes versus healthy. And this is really simple. It's a K-mer counter. I'm sure Genes is better, but I built one too. And I did consider cache coherence, by the way, when I built it. Nevertheless, what comes out of this is a K-mer matrix. You have your samples. You have the number of counts of each K-mer in that sample. On top of that, however, you use the sample metadata. And at this stage, you try to find K-mers that have a statistical effect or a discriminative effect between diabetes and healthy individuals. Once we have those K-mers, we can use them to capture reads, almost like a virtual hybridization. And there's a bunch of filtering and stuff here that goes on here that I can't get into, but what you end up with at the end is kind of an enriched read set, hopefully things that are relevant to your study question, and some significant K-mers that we group into what we call virtual probe sets. So these are K-mers that seem to be capturing these same things. Once we have virtual probe sets, we can do virtual hybridization using another software package. And this is just going to allow us to quantify those virtual probe sets in reads from additional samples. And at the end of that, we have a matrix again. We have signature quantities for some number of signatures and some number of samples. This is a data matrix, and the statisticians in the audience probably have a better idea than I do on the best ways to process it. So again, I know that was a lot, so I just want to go over it one more time with pictures. We have our 10 healthy controls and our 10 diabetics, the raw reads demultiplexed, going into Tagnostic. it's saying, has some settings related to case versus control analysis. We want to separate healthy from diabetics. And it's going to come up with a bunch of k-mers. That's these guys. And when we look at those k-mers and the reads that are associated with them, we can say, well, gosh, these k-mers are completely correlated in their levels across all the samples. They all seem to capture E. coli reads. We can make this into a virtual probe set that's kind of our E. coli probe set, and so on for B. vulgaris and Rhombautsia and other things that we discovered. Once we have that, we can take any read set, we can take these k-mer sets, send it through another piece of software, and it's just going to do a virtual hybridization. It'll do some normalization, converting things to parts per million and log transforming it so that you can better compare across different samples with different number of reads. But basically, you're just getting measurements of these signatures in every sample you send through here.
This is just 16S?
This is 16S plus ITS and 23S. So with the last few minutes, I'm going to show you a couple of results. We found a number of things, but I'll just point out a couple. So these are recapitulations of what's in the literature and what they found in their study, just as a sanity check for our method, because it is quite different. So on the x-axis, we have our 4 different categories of samples. Remember, with SigQuant, we did our discovery in 10 versus 10, but we can quantify all the samples. So this is all 47 of their patients. So we have our healthy, we have our kind of pre-diabetics, we have our definitely pre-diabetics, and we have our diabetics. And on this, we have a notion of the signature concentration in the samples, and it is just in log space so that you know. And as people have noticed, E. coli and Klebsiella are opportunistic pathogens. They tend to be found in diabetic colons as well as colons of people with other types of health conditions. Rhombautsia, this one seems to be a little bit more specific to prediabetes, but a number of publications have already pointed to this one as well. So, for this is sanity checking. But now I want to show you something a little more interesting. Here is P. vulgaris. On the left, it's a slightly different plot. We now have diabetics versus healthy, but this is just percentage of the microbiome. So in our diabetics, P. vulgaris is slightly enriched. It's about 20% of the microbiome, give or take. In healthy, it's 15, 16%. But this is certainly not a strong, exciting separation. But what happens when we look at our signature that's associated with P. vulgaris? You see that the levels seem to be going up slightly, and the error bounds become much smaller. So we're finding something that's really becoming enriched in the microbiome in diabetics. And is it P. vulgaris? Well, yes, it is. That's what our signature captures. The key is it's not all P. vulgaris. It's P. vulgaris that has a specific SNP in it. So here's a stretch of the 16S gene. This actually isn't the 16S, although we're capturing the ITS and all of that. There's a C versus A substitution here. And what our software is looking at with these probes is specifically the C variant. Our virtual probe set is tiling this region. So we wanted to figure out, is this meaningful? Well, what does it mean? So we went to GenBank and we found 10 high-quality P. vulgaris genomes that were completely closed. And it turned out 8 of them had the SNP in at least one of its 16S copies, and 2 of them didn't have it at all. So we sent those into a piece of software. Again, this is software I built that I don't have time to go into the details of, but I think you generally have the idea of signature finding software, right? We have one set of genomes, we have another set of genomes. We want to compare them and find out what's unique about this set of genomes. And what we found is that the C variant genomes contain some non-synonymous SNPs. There was about 20 kilobases worth of differences broken up into different regions. Some of them, unfortunately, were genes that have no known function, so I can't say much about those. But we did find some that might potentially be linked to diabetes. These 2 genes related to sugar and carbohydrate metabolisms had strongly conserved non-synonymous SNPs. And there was an insertion of a gene cassette. And this gene cassette seems to be involved in scavenging and metabolism of B12 from the environment. And the reason that's interesting is B12 is, in diabetes, many diabetics are B12 deficient. So there is potentially a biological link here. Now, I'm a computer guy. I'm not saying that this is biology by any means, but this is software that can at least see something that gives you a starting point. So to conclude, I've demonstrated a unique microbiome data analysis workflow that's enabled by 16S and ITS long amplicon sequencing. It's novel in terms of the informatics, and it's also novel because this long amplicon lets you see variations, and I accurately separate different strains of the same species potentially. You know, our approach, although it's quite different, does recapitulate many previously published results, so it passes the sanity check. We're further enabled to detect and explore strain level differences by finding probes that cover specific variants. And I will say with a caveat though about this particular study, by my standards, this is very small. There's a huge amount of variability in the healthy microbiome. And because of this, I would definitely want to see a lot more samples before I really believed what we were seeing here. And just final slide to conclude. Since Soren did ask for the bigger picture and open problems, I think what I wanted to put, I mean, the K-mer stuff that Gene brought up was fascinating. So all those methods could fall into here. I think faster 16S searching when people have helpfully filled your database with 10,000 copies of Salmonella could be interesting. But aside from that, bigger picture, DNA sequencing informatics, the whole time I've been involved with it, has been more evolutionary than revolutionary. Everything that we're doing today, a lot of the things that we're doing in sequence informatics are scaled up versions of what I was doing at Celera, what I was doing at 454, et cetera. But the technology itself has made a phase transition from being about generating small data sets on small number of samples to huge data sets on huge numbers of samples. And so I really think we should go back and instead of just saying, I want to make the next most fastest mapper, say, what should my analytical approach be, given now that we know so much more about processing big data and we are generating big data from these sequencers with tons of samples and millions of reads per sample. And that's my talk. Thanks.
Any questions?
Yeah, that would be.
So I thought the analytics part of the talk was great, but I was wondering, have you thought about comparing these results with a similar study, but with two groups of healthy people with different diets, as in assuming that the differences are actually there biologically, is that an association with the metabolic disease or some diet
Yeah,
difference?
absolutely. Unfortunately, their metadata didn't have detailed diet information, and definitely if you were to do the ideal study, you would include that. Yeah.
And just silly question, what's the aspirational or ideal case clinical or commercial, future of what you're doing?
Gosh, well, we're looking at two different areas. One is definitely clinical. We have some collaborators using this kit to try to help patients that have chronic inflammatory bowel diseases that haven't responded to other things to see if it's linked to the gut microbiome. In a way, this is almost kind of like these long shot genomics tests too that people do, but I think it's cool that they're trying, and I think it'll evolve slowly. The other thing we're looking at is clinical trials. So toxicology in particular, the way you metabolize the drug may be influenced by the microbiome. Thank you.
All right, so let me start by saying that I am so honored and humbled by those 2 days and the very generous talks and compliments, and it was an extraordinary meeting, and thank you all very much. It was very unexpected, but extremely pleasing. And I would like to start by thanking the organizing committee and both at Brown and then the colleagues who put this meeting together. And then I'll go to a PowerPoint and tell you a little bit about reflections with the occasion of this 70 years. So let me start by saying that we had an extraordinary team that organized this meeting, and I am extremely grateful to them. And I'm going to start by calling Susan Alden to come in front to get a little gift. And thank you for
Oh, thank you.
for amazing. Susan is she's also my assistant in the department for many years, so she's extraordinary, outstanding. Thank you so much.
Sure, my pleasure.
Second, Meg, an extraordinary member of the team. Thank you for everything you did. It's amazing, amazing work. Thank you so much. And I have one for Jesse. I'm not sure if he is here today.
No, he's not.
He's not, so he will have homework. We'll do this. So now I have some gifts, brown gifts for the organizing committee, just to recognize them. Just a smaller little gift. Ooh. So let's start with Bjarni, the chair of the committee. Thank you so much. Amazing, amazing. Russell, the second member of the committee. Thank you so much. Outstanding work. And Derek, my best student ever and an inspiration. Thank you so much. All right, so I want to thank now the departments, the two units that sponsor this event. One is my Department of Computer Science, and I'm going to highlight the fearless leader Roberto, Tomasia, our chair, and the department manager Lauren Clark. You guys are amazing, an inspiration. Thank you so much. I want so to mention the CCMB and the 20th anniversary of this year, and I hope at the dinner tonight we'll have a toast for 20 years of computational biology at Brown. But the leaders of the center who are exceptional and inspiration was Sohini Ramachandran. She is the director of the Data Science Institute and former director of CCMB, and Dan Weinreich, who is the present director. So let's acknowledge their work. So a third pillar of this support came from President Paxson's office, and I want to thank her for her remarks yesterday and a lot of support over the years and for this particular one. Now, there is another missing gift for Vinit Bafna, the fourth member of the organizing committee. He had to leave this earlier, but he got his present. All right, so I'm going to start with my family, who has been my greatest support over the years, and some of them are here. The babies couldn't be our grandkids, but they are somewhere on Zoom, I'm sure. Oops, and there is the entire family that I want to thank them for everything and the love and the support over the years and so on. So I'm going to talk about some of the people who inspired me. Just a few minutes of several, and I will have a conclusion of the thoughts that came to me at this moment at the 70th birthday. So I wrote a number of essays about my interaction with some of the big names I was privileged to work with them closely, or scientists who inspired me through their work. And the essays were written mostly for my students to share lessons that I learned from those great collaborators, mentors, heroes of mine. So I'm going to conclude my remarks with the axioms, which I call those lessons, I call the axioms of von Neumann and the others, with the axioms that are my reflections of me at 70. So I'm going to go and pick some of the axioms from the heroes and so on. So since graduate school, I was fascinated by von Neumann, and in some small measure, I worked on problems in computer science, a little bit in physics, a little bit in biology, a little bit in mathematical economics. So I am trying to follow the von Neumann set of seminal themes that he worked in his seminal research. So I have just a few pictures of him, but I have his axioms that I put in my essay. So I wrote the essay, and this was the first day when I joined Brown. They asked me to talk about my journey to Brown. So I used John von Neumann, and I said, in search of von Neumann, randomness is beautiful. So here are the axioms, the lessons I learned from him. Randomness is beautiful, work on the hardest problems, continuously search for teachers. Scientific teams are fragile. A crisis is a terrible thing to waste. And in the end, the love you take is equal to the love you make. My other hero, which actually I met in Rhode Island, of all places in the world, at Salve Regina University. When he emigrated to US, he invited me to come and spend the day with him there. And it was a memorable day. And I wrote essays, 2 essays about him. As you know, he was extraordinarily critical, but eloquent, and the ultimate professor of programming. So when I was in Romania as a graduate student, he was one of my heroes. So from my essay, you have a few pictures. Oh, oops, there is a correction. Sorry. So in my essay, portrayed him as the Don Quixote in his quest for perfection in programming, and honesty and eloquence, and encouraging us to write, and beautiful mathematics. So his axioms, in my view, were: life is the same as mathematics. Computer science is mathematics plus Murphy's law. Beauty is our business. We need to write programming at such a simplicity and elegance, because only then they are reliable. Simplicity is a prerequisite for reliability. So Eric Davidson, among all of them, is probably one of the most sentimental in the memory that I have. I worked with Eric for 15 years. He was my postdoctoral advisor, and we collaborated and did a number of extraordinary papers. I spent time over the years at Caltech, and changed my life, one of the people. So he was my closest friend, mentor, professor, everything. So it was an extraordinary experience to learn from him. Again, the toughest scientist you ever hear, the toughest critic, but always in the most professional way. And I miss him very much. And you could see here me at Caltech when I got the Brown offer. So I have to say that Eric is the only prophet that I know. He was predicting the future. So when I was starting interviewing, he predicted I will end up at Brown. When the presidential election long time ago was with 20 people, Obama won, but at the very beginning, nobody knew all those 20 people. He told me that either Obama or the New Mexico governor, what's his name? Richardson will win, and we indeed Obama. And thank you, Craig, because you took me once at the birthday of Richardson with you, and we want to say happy birthday, sing happy birthday to him. So it's now he passed, so it's a great memory. Thank you. So here is Davidson at Brown. Stu Gieman is having fun with him. Here is again giving a phenomenal lecture at Brown. What are Eric Davidson axioms? Very meaningful to me. Serious people do things seriously. He was a tough guy. The answer on how to recognize the right place through the wall of thorns lie in the shape of a mental triangle composed of intuition, current knowledge, and logic. Logic rules. This is another beautiful axiom. If we care about anything outside our current events, we need to care about those who will carry the brilliant torch forward. What a beautiful. Life is short, keep in touch. Those are things. Use it or lose it, the biology principle. And this is my most, the one I love the most. Have inexhaustible optimism, inexhaustible curiosity, inexhaustible energy, and inexhaustible honesty. That was Eric. And in the end, the love you take is equal to the love you make. Solomon Marcus, my PhD advisor in Romania, mathematician of stature and computational linguist, was extraordinarily influential for me, inspiring me. He gave, after a big surgery at his stomach, taking a part out of it, he came to Brown, and I said, send me an abstract. He sent me 8 abstracts for 8 lectures. And he gave those lectures, and we have them, and one day I'll write a book, but they are on my webpage. And we had a tremendous time together. This is John Conway. When we went to Romania, Professor Marcus was a big fan of Conway. So here they are. This is my former assistant, Aaron. Here we are at Brown. Craig, this is our map, which is on the second floor. We should visit. We have the Celera gene map. So that was an extraordinary time. It's interesting. He prepared those lectures by writing on the transparencies. He was on the old-fashioned. So, I'm thinking of using some software to collect the text of his lectures. There will be no problem like you have PowerPoints to be distracted by this. Everything was written on the slide and he was reading. It's exactly what you say to your students. Don't write that many things on one slide. No, he was just like this. So he was a mathematician of great stature. In fact, due to his PhD advisor and the European advisors of many, I have 9 links to Euler because of, if you just go through the advisor of advisor, and I think 11 to Leibniz because of the great mathematical school that and advisors that he had. So what are Solomon Marcus axiom? Be a language theorist. Be a first-class scientist in at least one of the disciplines of your interdisciplinary research. Be a mathematician of the continuous and discrete. be an intramath, intersciences, and cross-culture scientist. So he was, you know, inspiring us to do all those expansions of what we're doing. Be a guardian of high standards, know a lot, really a lot of mathematics. And he said, be a storyteller. And he claimed that the part of Romania we are coming from, the Moldova part, is very rich in storytellers, so we cannot escape. My next hero is John Conway. So John Conway we met at Sandia. We started collaborating, and he was the guest of honor to my son Larry's bar mitzvah. And he used to challenge Larry with various games and so on. So my kids, my children, my boys brought to me access to some great mathematicians, as you will see, and I want to thank them, both of them, for this. So John Conway, here is Larry at his bar mitzvah, and John Conway, guest of honor with games. He was challenging already Larry to play his games, and it was, here is at Brown giving us lectures. Here is Stu Gimman introducing him. Here we are traveling to Romania. He is in a wheelchair in the first class. I am in the second. And we went because he was given an honorary doctorate from my alma mater. So you'll we'll see a little bit more about him. So I have a unique tape. His wife complained to me that he has a beard that is terrible. And with the occasion of the Romania honoris causa degree, he decided to get rid of his beard. So we have this very interesting picture in which he became 20 years younger looking, and was a wonderful experience. We became quite close friends. So I have, and it's going to be on my webpage. His wife thanked me very much for this. Here is him without beard. We are together in his wheelchair, and him getting the honorary degree from University of Iași and giving 3 amazing lectures there, all recorded, we have them. And here is him with short pants, ready to play in his wheelchair, ready to play games with the students there immediately. That was his passion for playing game with the student, teaching all the time and challenging us. An amazing person. He died during COVID, unfortunately. Ken Arrow, the other hero of mine, his parents were born in Romania, like mine. So when he was invited here to give a von Neumann lecture, we discovered this, and then later I invited him to go to Romania to get a degree from there, and he was extremely excited to do this. So here is him at Brown. You see Eli Uffol here and Leon Cooper in the back and Stu Gimman. And guess what? Eric Davidson. We were at the Brown Symposium with many distinguished von Neumann lectures. Here we are in Romania now with him. And he gave us some beautiful lectures on economics. One of them talking about how the future influences the present. Anyway, can you just hear a little bit his voice would be very nice.
It doesn't protect itself so well. Here, let's say the United States and the United Kingdom, one of the big problems where they had a lot of lending on mortgages, and the mortgages in turn were paid for by issuing securities backed by these mortgages. And we have a very rapid rise in housing prices. So people were buying houses feeling extremely safe because they can sell.
All right, just a little bit of And we have two phenomenal lectures that he gave to Brown. So we are very fortunate to have him recorded. The second one was how the future affects the present from an economic point of view. The next one is Paul Erdős, the mathematician of fame, the most prolific mathematician, 1,500 papers. And I got to know Paul Erdős because of my son Lee, who we were living in New Mexico at that time, and he was the finalist of the Mathematical Olympiad in New Mexico. And one day he says, "Dad, there is some speaker that is coming. His name is Paul Erdős." So I said, "What?" So I was at Sandia Labs at that point. So I went to University of New Mexico and I said, "Can I have him to give a talk at Sandia and so on?" And the chair of the department said, "Do you know what you're doing? You have to sign for him and to bring him in one piece. He doesn't know anything. He doesn't have any money. All he does is mathematics. You take a big responsibility if you want him." So we went and he gave 3 talks, one at UNM, one at Sandia, and one at the finalists of the Olympiad, exactly the same talks. So very interesting. So also my son, because of the youngest son that we had, filmed the whole thing. Was a party at our house with Erdős. So we can look at something very unique, which is going back in memory lane. And this was a meeting with Ernie Brickel's group where I was there, and Ernie is going to show up here, and here is Ernie, as usual with his argumentation was giving us, and Erdős was on the phone. Here is me, excited about many things there, and I know that the kids were running around. Let's see if I can get more things to see Erdős. Then again, it's going to be on my webpage, and we have various colleagues from Ernie's group. 2 of them are professors at University of Oxford. Here is our Leslie Goldberg, and definitely a beautiful all right, our next my next friend, and in fact I consider him like an academic brother, Alberto Apostolico, who passed away so young, and his wife Titi is here, and we love him very much, and I give my students his papers on suffix trees as a required reading. And here is Alberto and Titi in our conference room on the 2nd floor where the Human Genome Map is, when he gave a wonderful lecture. Here we are at Ischia at a conference where he invited me to give a talk, and I think it was the last time I had the pleasure. And here is Alberto and Professor Franco Preparata, who is the visionary founder of our Center for Computational Biology, and gave a talk yesterday about the beginnings. Here we are at the conference in Venice, where we celebrated the memory of Alberto. So you will see we started, Mike Waterman, Pavel Pevz, and myself, the conference RECOM. And for the 10th anniversary, I said, there cannot be a better place to go but to Venice, and Alberto and Titi were organizing the conference. And was the first time when the steering committee came with the wives at the meeting, and I made a contribution in that way. Here are Alberto's axioms. He was a really fun guy, in love with algorithm, champion of rigorous methods. And he taught me an Italian phrase, transina facende, which means very, very slow. So I have an axiom, time is short. Intellectual honesty leaves us no time for Fracina facende. He used humor as a critical commentary, a great storyteller. My son Larry used to retell his stories. And in the end, so I have written an essay about Mike Waterman. Mike Waterman is here, and I am so delighted. I hope he is here. Yes, Mike? So Mike is an inspiration for me, and we have been working since Sandia time. And I wrote an essay about him, and I put the axioms of Mike Waterman here. So intertwining continuous and discrete mathematics is quintessential to computational biology. Be a world scientist in at least one area of your interdisciplinary research. Be an intramath, intersciences, cross-culture. Mike is also writing literature, memories. Be a guardian of mathematical rigor. In my collaboration essays writing, I had an observation that all the people I admire are extremely tough critics. So I said, you know, it's a style how a person deals with criticism. But Mike is an exception. If you can't say something good about somebody, don't say anything at all. And this is also von Neumann axiom. He was exactly like this. His daughter was here giving a lecture and told about it. His approach to criticism. And I like this axiom very much. You can know a person by his heroes, or his or her heroes. So that was Mike. And of course, my typical ending from the Beatles. We're almost done. Julie and Gwen Brown. So when I joined Brown, I was given the chair professorship in the name of Julie and Gwen Brown. It's called Professor of Computational Mathematical Sciences. She was the donor of $20 million to start the Center for Computational Biology, and part of that was a chairship in her name. So for many years, I think I was the only chair at Brown having a businesswoman name. So Julie was an inspiration, and I used to give talks especially to female students to take the example. So she came from Vietnam, born in Vietnam, educated in America in computer science and engineering, going to Ford Motor Company and raising through the ranks to vice president, quitting the job, starting a new company, becoming billionaire, and donating money to Brown because her two children were educated at Brown. She was on the board of trustees and so on. But she was a very low-key CEO, not always on TV, liking to do things behind the scene, a force for good, but not to be seen all the time doing it. So for a while, she was the minority owner of the largest company in the US, providing automotive parts for all the American car companies. Her company built the interior design of the cars. I have only one picture of her. Here is Julie and Gwen Brown, an inspiration. So Bob Zimmer just passed away. He was the provost when I got hired. He called me to attract me to come to Brown and promised to me a chair professorship. And when he said that, I said, I want to be John von Neumann professor. And he said, it's not that simple, but we have a chair professorship, so don't worry, and so on. So once I joined Brown, in the first week, he said, Soren, we have to work together. Marine Biological Laboratory, I know that you and Eric Davidson were there. We need to strengthen collaboration with them and some DOE labs. So I was starting working with him, and it was very nice. A year later, he is appointing me as the new director of the Center for Computational Biology. An extraordinary person. I don't have much about him, just a picture, but Brad Stevens, my favorite columnist at New York Times, called him the America's best president. So he returned from Brown. I don't know if I was the reason, but after a year, he went back to Chicago to be the president. So I admire him very much, and he was the most enthusiastic sponsor for computational biology at Brown. How am I doing? Okay. So I want to say a few things now, getting to the ending. So in '97, Mike Waterman, Pavel Pez, and myself started the RECOM conference. And here are the pictures of the people participating. So this is a really historic picture. This is Nick Metropolis and Bill Byers. Nick Metropolis and Dick Byers. They attended the first RECOM when Mike and Pavel and me were in Santa Fe, and some people might still remember that conference. Here is Mike and Bill Byers, and this is Ulam's daughter, Ulam's granddaughter. Good, Mike, good. Here is the table in which you see a number of interesting characters that we recognize of fame. Metropolis, Big Byers, Rich Roberts, David Botstein, Martin Karplus, Pavel Pevsner, and the DOA guy. What's his name? I forgot.
Yeah,
Bransko. Okay. So here is Mike, the chair of the first computational biology conference at El Dorado, Santa Fe, giving the remarks of the starting of a new field, computational biology. Here are more pictures from that time. Here are the founders of RECOM, a memorable picture. So with that occasion, Mike took Pavel and me to visit Francois Ulam, Ulam's wife that was still alive. So that was a memorable visit. And that was like a rite of passage to a new generation. And here we are at the conference, and that's what it is. So my next section says, thank you, Eric Davidson, Craig Venter, and Mike Waterman for bringing me closer to John von Neumann. So I think you guys are the three mentors and professors and inspirations for me, but in some fundamental way, you brought me closer to von Neumann, and I want to thank you for that. So first, Eric Davidson. I worked 15 years with him, and one of the papers we wrote was at the 50th anniversary of John von Neumann, The Computer and the Brain. We wrote a paper about, with the same style of writing as John von Neumann, a comparison one by one of the components from the brain and the electronic computer, how the two materials, the two principles are mapped to each other. And we did the same with the regulatory genome, how genes are regulated, how is the computer information processing system there. So in doing this, I observed that Eric was just so excited about von Neumann, couldn't stop reading von Neumann. So it was a spectacular understanding of his work and talking about. So Eric brought me closer to von Neumann. Craig, who hired me at Celera, when I got there, I was coming from Sandia on a military base, New Mexico. Manhattan Project was in the air also. So I couldn't but think, oh my God, I have a chance to work on a similar bigger than life project, which was the Celera Genome Project. So all the time, this was on my mind, what once in a lifetime I have. So in this way, Craig, you brought me closer to von Neumann. Thank you. So I have a story to tell about Craig. So story about Craig is this, the interview with Craig Venter. I have a little story. So Gene Myers invited me to come to interview, and there was a 2-day interview, and was a wonderful set of colleagues there. And Gene said, listen, I know it will be lovely to have you, but if Craig doesn't like you, it's not going to work. So here I am the first day with the team, and then the second day in the morning, I am going to Craig's office. And who do I find them? Paul Gilman, the vice president for PR, the former chief of staff of Senator Dominici from New Mexico, was the biggest fan, the biggest supporter of genomics in New Mexico. And Paul said, look, I was following you. I just saw your science editorial with the 3D icing model. We're proud of you as a national lab scientist, and we're delighted that you're coming here. And all right, so I said, friendly reception. We talked about New Mexico, and and then something happened. The door opened, and Shadow, Craig's dog, the first dog in genomics, because this amazing creature was sequenced, and we have the genome of Shadow. So Shadow comes, and I was waiting to see Craig, but it took a few minutes till Craig arrived, but Shadow was there. And this reminded me of a Romanian fairy tale in which there was a Prince Charming that had this kind of weapon, and the weapon would arrive first, and then the prince would... So I said, this is where, all right. So finally, Craig comes, and it was the best interview ever in my life. At that time, I was getting a pharmaceutical company wanted me to hire me as a director of bioinformatics, and they were talking to me as computer people. So then I asked Craig, what is the role of algorithms at Celera? And Craig, without a hesitation, said algorithms are the make or break of Celera. So at that time, I knew that this is the place to be. So he made me an offer I couldn't refuse. 2 weeks later, I was there. Even my family, who was trying to find a job for Gabi, they said, go, it's clearly the bestest chance for you to go, and so on. So this is my story. Thank you, Craig, for bringing me closer to von Neumann. So now, Mike, maybe you brought me closer, much closer than all of the people, because over the years there were so many interactions with Ulam and so on. So I want to say something about Bill Byers. So Bill Byers was the one who brought Mike Waterman to Los Alamos, where Smith-Waterman was created, the famous algorithm. And Mike shared with me one email from Bill. And this is what Bill says. I was indeed honored by compliments from your colleagues. He was visiting USC. Your friend, now our friend Sorin, was so kind to me. His remarks about von Neumann to Ulam to Bayer to Waterman to Sorin was the highlight of my life. So Mike, thank you so much. You brought me closer to von Neumann. So now I'm going to tell you a few of the axioms that I actually picked from my mentors as a way to end this presentation. So the first axiom is another hero of mine. If you guys come and visit my office, you're going to see a very big poster of John Lennon. There are no problems, only solutions, he says in a beautiful song. Next is Craig Venter axiom. I'm hoping I'm capturing the spirit. Algorithms are the make or break of genomics. Is this approved? Absolutely. John von Neumann axiom. Work on the hardest problems, at the same time on the most theoretical and the most practical. That's what he did. And we look at him as an inspiration. Eric Davidson, you already saw that, has inexhaustible optimism, inexhaustible curiosity, inexhaustible energy, and inexhaustible honesty. Alberto Apostolico axiom. Time is short. Intellectual honesty leaves no time for transina faccende. And finally, another Eric Davidson axiom. If we care about anything outside our current events, we need to care about those who will carry the brilliant torch forward. And at Brown, I have exactly 3 names: Derek Aguiar, Vitambra Singh, and the incredible Sohini Ramachandran. And I believe they are going to carry on. Thank you very much.